Hello champs, I hope you all are doing great. My name is Biswajit, I'm your botany teacher. Today we are going to discuss a very important, conceptual, interesting chapter of class 12th, and the chapter is Principles of Inheritance and Variation. This chapter is very important for NEET point of view, and I have seen many students facing problems to solve questions from this chapter. The reason is very simple, because they do not have proper understanding of this chapter. I assure you, if you watch this lecture honestly with dedication and concentration, you won't face any problem to have proper understanding of this chapter. You will be able to solve any type of questions from this chapter. So the topics which we are going to cover today are various terms related to genetics, Mendel's laws of inheritance like law of dominance, law of segregation, law of independent assortment, and the exception to these laws like incomplete dominance, codominance, and linkage. We will talk about chromosomal theory of inheritance also. I have not written here, but I will discuss that. Different methods to determine sex of various organisms, mutation, genetic disorder, and we will also talk about pedigree analysis. Pedigree analysis and chromosomal theory of inheritance I have not written here, but I will discuss that with you. So when I say genetics, two things you have to remember here. One, the first important thing is, who is father of genetics? It is the great Mendel. Mendel did a tremendous, wonderful work on pea plant, and unfortunately his work was never recognized or appreciated by any of the scientists of his time. So you might be doing some great work, but it is not necessary that the people will come and recognize and appreciate you, but you have to keep on working, keep on doing your good work. Okay, the second thing what you have to remember here is, who was the person to coin the term genetics? It was Bateson. Father of genetics, Mendel. The term genetics was given by Bateson, and this genetics includes two different things. The first thing is heredity, the second thing is variation. So what is heredity and variation? Before understanding that, we must know something about character. I will discuss with you just after a few minutes in detail about character, but as of now, you should know one thing, that the character may or may not be controlled by gene. Let me give you an example. When I say drinking of wine, this is a habit, this is a character. Okay, and this character is not controlled by gene. But when I say skin color or height, these are characters and they are controlled by genes. And that is why I will say a character may or may not be controlled by gene. Okay, and gene is basically nothing but DNA, and this DNA are present in the cell, in the nucleus, as well as in the various organelles like mitochondria and plastid, right? And whenever the organism reproduces, this new cells will be formed. That means the DNA will be transmitted from the parent to the progeny. And when the DNA or the gene gets transmitted from the parent to the progeny, so the characters will also get transmitted. And this transmission or the transfer of genetically based characters from the parents to the progeny or offsprings is called as heredity. And because these characters have transmitted from the parent to the progeny, that is why the parent and the progeny, even the progenies, they will show some similarities with each other. And the similarities between parent and progeny or among the progeny is due to heredity. But you might have noticed that you are not exactly identical with your parent, neither with your mother nor with your father. You are not exactly identical with your brother or sister. There is exception, identical twins, but except identical twins, you are never, no two brother or sisters, they are identical to each other. Okay, and this degree of differences or dissimilarities between the parent and the progenies and among the progenies is called as variation. So in genetics, we are going to discuss the similarities, and we are also going to discuss about the differences or the dissimilarities. That means we are going to discuss about heredity and variation. There is another term which resembles with the heredity, and what is that? That is inheritance. What do you understand by inheritance? This is the process of heredity. That means this is the process of transmission of genetically based characters from parents to progeny. Okay, So now let's talk some of the important terms related to genetics. The first term which we are going to consider here is the character. So any morphological or physiological feature of organism which can be measured or observed or both, they are called as character. So when I say morphological feature, when I say morphological feature, I can give you one example of morphological feature which will be an example of character, and that is the height. That is the height. So when I say height, that means this is the character and this is the morphological feature. And when I say physiological feature, this is also going to be character, and I can give you one example, that is the ABO blood grouping. ABO blood grouping. Okay, and this is also a feature, physiological feature. So whether the feature is morphological or physiological, if the feature is well defined, then I will call that as character. And these characters are controlled by gene and environment. For example, skin color. No doubt skin color is controlled by gene, but it is also affected by the environment, the humidity, temperature, etc., etc. Right? Now, what do you understand by trait then? Every character has some alternative forms, and those alternative forms of character are nothing but trait. For example, if I say height of pea plant, that is a character. Okay, if I say seed shape in pea plant, that is also a character. Okay, and these characters will have some alternative forms, and those alternative forms are called as traits. So when I say height in pea plant, this is character, and the traits or the alternative forms of this character is tall plant and dwarf plant. So tall and dwarf, they are the traits of which character? Of height. When I say seed shape, this is a character. So what are the alternative forms of these characters? Round seed and wrinkle seed. So these round and wrinkle seed, they are the examples of traits. When I say flower color in Mirabilis jalapa, which is also known as 4 o'clock plant, okay, its color, the flower color is character, and the traits will be red, white, and pink. When I say ABO blood grouping in human, so ABO blood group is character, and the blood group A, blood group B, AB, and O, these are the traits. So from here you can conclude that you can conclude in these two cases. In these two cases you can see one character, two traits, one character, two traits. But in these cases you can see one character having more than two traits. Here 3 traits, here 4 traits. So I can say, that is why I can say that one character will have 2 or more than 2 traits. It will have 2 or more than 2 traits. Okay, so any physiological or any morphological well-defined feature of organism is called as character, and the character will have some alternative forms, and those alternative forms are nothing but the trait. Okay, now let me tell you something about gene. Okay, so any chemical which contains gene is called as genetic material, and with respect to cellular organism, the genetic material is always DNA, and the DNA is always double-stranded. It can be circular, it can be linear, but it is always double-stranded DNA. So I will write here one point, that the genetic material with respect to cellular organism is always double-stranded DNA with respect to cellular organisms. All the cellular organisms, they have double-stranded DNA as genetic material. Okay, so suppose this is a double-stranded DNA. So suppose this is a double-stranded DNA, and I have taken various segments here, segment A, B, C, D, E, like this. Okay, suppose this segment A has the information to form the tRNA. So this segment A will undergo the process of transcription to form the tRNA. So I will say the segment A has the function of producing tRNA. The segment B, it does not have any function. Segment C has the information to produce the rRNA. So it will undergo transcription to produce rRNA. So I will say the segment C has the function to form or to produce the rRNA, ribosomal RNA. And The D segment, it does not have any information, so it is not going to form any kind of RNA. And this segment E, it has the information to produce the mRNA, so it will undergo transcription to produce mRNA, and the mRNA will undergo translation by the help of ribosome to produce the protein. Okay, so here you can see the segment A, C and E, they have some functions. Okay, so I will say A, C and E, they are the functional segment of genetic double-strand DNA, and double-strand DNA is nothing but the genetic material. So any functional segment of genetic material, that is gene. So I will say in this case ACE, they will be genes. Segment ACE, they will be genes. But B and D, they do not have any function, that is why they are not genes. But so what do you call them then? See, this B and C, so B and D, they are present between genes. Here A and C, they are genes. So B is present between genes, so I will say this is intergenic sequence. Here also this D is present between genes, so this will be also intergenic sequence. Okay, so I will say here I will write here they are not genes. So you may ask me one thing, sir. So gene means any structure, any segment of DNA which produces RNA, that will be called as gene. No, this is incorrect. Why? Because there are certain sequences of DNA which do not produce any RNA, but still they are called as gene. For example, promoter. It never produces any kind of RNA. It is simply a binding site for RNA polymerase and various transcription factor. Silencer, it does not produce any kind of RNA. Okay, or enhancer, it does not produce any kind of RNA, but they have some function. So any structure of any segment of the DNA, whether they are going to undergo transcription to produce RNA or not, it does not matter. But if they have any sort of function, then those functional segment of genetic material or double-strand DNA will be called as gene. Okay, so whenever I say gene, you have to think with respect to which type of organism? The law organisms, and that is why you have to think about DNA. Which type of DNA? Double-stranded DNA. Clear? Very good. Now, just like character, this gene also has different forms. The different forms or alternative forms of character are called as trait, and the different or alternative forms of gene are called as allelomorph. And in short, we call allelomorph as alleles. Okay, and alleles are nothing but the different molecular forms of gene. Okay, so when I say different molecular forms of gene, that means there will be some differences in the base pair sequence in the DNA. Let me give you one example. So suppose this is one double strand. Just a minute. Suppose this is one double strand DNA. Okay, and suppose this is another double strand DNA. These are two double strand DNA molecules. Okay, suppose here G A G sequence is there, so it will be C T C, and here G T G is there, and here suppose it is C A C. Okay, and in this case all the remaining sequences, that means the sequence present over here are exactly identical to the sequence present over here. The sequence present over here will be exactly identical to sequence present over here. There is little differences between the two DNA molecules, and that is why I will say these are the two different forms of the same DNA. There is some difference, very little differences between these two DNA molecules. So I can say that these are the two different forms of the same DNA. That means there is very little differences between these two DNA molecules. Okay, here I have AT base pair and here I have TA base pair. Okay, and these are called as alleles. These are called as alleles, or different molecular forms of this DNA molecule, and different molecular form of the DNA or gene is called as basically allele. Okay, and this is I can say this, suppose this is the normal beta gene forming beta chain forming gene, and this is sickle cell hemoglobin forming beta gene. Okay, so the gene controlling the height of the pea plant has two different forms. One is capital T, another is small t. Capital T, small t, they are same gene, but they are different forms of the same gene. Okay, these are capital T and small t, they are one gene, but they are different form of the one gene. And in case of human being, the ABO blood grouping is controlled by one gene, but this one gene has three different alleles or three different forms. One is IA, second is IB, third one is I, or I can say I naught. So here the gene for controlling height of the pea plant is one gene that has two forms, capital D, small t. So one gene and alleles are capital D, small t. Here the gene controlling the ABO blood grouping in case of human, one gene I am having, and that one gene will have three different alleles. So I can say, that is why I can say one gene may have two alleles. Here I have how many alleles of the one gene? Only two gene, but here I have three genes. So I have three alleles. Okay. So I can say one gene may have two or more than two alleles. Here one gene has two alleles. Here one gene has three alleles. So I can say one gene may have two or more than two alleles. And whenever one gene will have more than two alleles, that is called as multiple allelism. What do you call that as? You call that as multiple allelism. So let me write that part here. Okay. So what do you understand by multiple allelism? Multiple allelism is nothing but the phenomenon in which a single gene will have more than two alleles. It will have more than two alleles. And the examples are, one of the example is the gene controlling the ABO blood grouping. The second example is the gene controlling the self-incompatibility. Self-incompatibility. Okay. So let me write here examples. I can say gene controlling ABO blood grouping, and another example I can say gene controlling self-incompatibility. You might have heard about gametophytic self-incompatibility, sporophytic self-incompatibility, right? So one gene will be responsible for that, and that one gene will have multiple alleles. And existence of multiple forms of a given gene, that is multiple alleles of the same gene, that is called as multiple allelism. And remember one thing, if the organism is haploid, then only one allele will be present. If the organism is diploid, then two alleles will be present. The alleles may be same or may be different, but two alleles will be present. If the organism is triploid, then three alleles will be present. If the organism is tetraploid, four alleles will be present. Okay. So when two or three or four alleles are present, they may be of same type or different type. Okay. So let me write that part here. Okay. You can write that part. Okay. So that if the organism is haploid, every gene will have only one allele, or I can say every cell will have only one allele of every gene. And if the organism is diploid, every gene will have two alleles in each cell. The alleles may be same or different. If the organism is triploid, then every gene will have three alleles. The alleles may be same or may be different. Okay. Now, the next term which we are going to discuss is locus. The plural is loci. So what is locus? We know gene is nothing but DNA, and DNA is present in the chromosome. So the position of the gene on the chromosome, that position is called as locus. Okay, so this is a chromosome, this is also a chromosome. Suppose these are two homologous chromosomes. Okay, so suppose this is chromosome number 5 and this is also chromosome number 5. Okay, so these are homologous chromosomes. Okay, so you can see here, so these are chromosomes. So let me write here, suppose this is chromosome 5, this is also chromosome 5. So these are homologous chromosomes. So let me take one position here. So this is one position, and the same position I am taking here. So these are the same positions. That means these are the same locus. Okay, and suppose here I am having an allele capital A, and here I am having the allele small a. So I can say here I have taken one locus, because this one and this one both are same locus. One locus, but allele is different, capital S and small a. Gene is one, because capital A and small a, they are the alternative forms of same gene. So I will say here, this is indicating, this one is indicating what? This one is indicating one gene, one locus and two alleles. Okay, suppose these are homologous chromosomes. Suppose this is also chromosome 5, this is chromosome 5, this is chromosome 5. Okay, so these are homologous chromosomes. Okay, suppose let me take some locus here. So this is one locus, and the same locus I'm taking here also, and these are the same locus. Okay, suppose here I have taken allele S1, here I have taken allele S2, and here I have taken allele S3. Okay, so I can say how many locus I have taken? One locus, because these three are the same locus, they are representing the same locus. Okay, so I can say here I have taken one locus, and how many genes? One gene, but how many alleles? Three alleles. How many alleles? Three alleles. And that is why I will conclude here that one locus will always have one gene. Here I have taken one locus, here also I have taken one locus. How many genes? One gene. Here also one gene, and that is why I will say, I can say here that one locus will always have one gene, but the number of alleles can be 2 or it can be more than 2. It can be 2 or it can be more than 2. Okay, now let me discuss about the alleles, different types of alleles. Okay, so suppose there is an allele which is wild allele. So what do you understand by wild allele? The allele which is present more frequently in a population, that is called as wild allele. I repeat, those alleles of a gene which are frequently found or more common in a population, they are called as wild allele, and those alleles which are less common in a population, they are called as mutant allele. Okay, so this is a wild allele, and this wild allele underwent modification. Okay, and the modification is nothing but change, and change is called as mutation. So what happened in the allele, that means in the gene, modification or mutation took place, and because of mutation, new allele is formed. And because this allele is formed due to change, that is due to mutation, that is why this allele will be called as mutant allele. Okay, and this mutation, because this mutation is nothing but the change or modification, that is why this mutant allele will be called as modified allele, and this wild allele will be called as normal or unmodified allele. Okay, and this wild allele is normally dominant in nature. It is not always dominant. The wild allele is normally dominant, not always dominant. And the mutant allele or the modified allele is normally recessive. It is not always recessive, it is normally recessive. I repeat, allele is different or alternative molecular form of gene. When I say alternative molecular form of gene, that means the DNA will have some differences in its base pair, little differences in the base pair. Okay, they will be called as alleles, and the alleles are present on the chromosome, and the locus or the position of the alleles or the genes on a chromosome are called as locus. Okay, and the allele, if it is more common in a population, that is wild allele, and if it is less common in a population, that is recessive allele, mutant allele. Okay, and this mutant allele is formed due to the mutation or the modification in the wild allele. Okay, so let me write one point here, that wild allele is more, you can write here, wild allele is, let me write here, wild allele is more common in a population. So those alleles which are more common or which are found more frequently in a population, they are called as wild allele, and those alleles which are less common or which are present less frequently in a population, they will be called as mutant allele. Okay, now, so whenever the mutant allele is formed, the mutant allele is formed due to mutation or change. So whenever there is a change or the mutation, the allele will have three fates. three different types of fates. Okay, it may have four fates, but as per NCERT, it will have three fates. So what are the fate or the result of the mutant allele? The mutant allele may not produce any enzyme or any protein, or it may produce protein or enzyme, but that will be non-functional, or it may produce normal enzyme or less functional enzyme or protein, or it may form a completely new enzyme or protein with entirely different function. Okay, so let me repeat. So whenever there is a wild allele, it will produce normal protein, normal enzyme. But when it undergoes mutation, the mutant allele may produce protein or may not produce protein. And if the protein or the enzyme is produced, it may be non-functional, it may be less functional or normal, or it may be entirely different enzyme or protein. Okay, so the fourth one is not written in the NCERT. Okay, so if the mutant allele produces no protein or no enzyme, or if it produces no protein but the protein or enzyme is non-functional, in that case the phenotype will be affected. In that case, what will be affected? The phenotype will be affected. So I will write here. So in both of these cases, what will happen? The phenotype will be affected. Phenotype gets affected, or the phenotype gets modified. Gets affected or modified. You can write affected or modified, no problem. Okay, and whenever there is no protein or enzyme due to mutant allele, or whenever there is non-functional protein or enzyme due to mutant allele, the phenotype produced will be recessive phenotype. Which type of phenotype will be produced? Recessive phenotype will be produced. But if the mutant allele still produces the normal enzyme or protein, or little less functional enzyme or protein, in that case I will say the phenotype will not be affected, the phenotype will not be modified. Okay, so I will write here that point. Okay, so phenotype does not get affected or modified. Okay, and in this case the phenotype which will be produced by the mutant allele will be dominant phenotype. Okay, so these are the points you have to remember here. Now let me tell you something about chromosomes. Okay, so here I am going to tell you something about the types of chromosomes. The first thing you have to remember is the chromosome can be somatic chromosome, or can be not somatic, autosomal chromosome or sex chromosome. So what do you understand by autosomal chromosome? Those chromosomes which have genes which are responsible for controlling the somatic characters only, they are called as somatic character, autosomal chromosomes. But if a chromosome has genes which are controlling the sexual characters also, they are called as sex chromosomes. Here the important point you have to remember is that autosomal chromosome will have genes which will control only the somatic characters, but sex chromosomes, they will have genes which will be controlling somatic characters, and some genes will be controlling the sexual characters. Okay, that means the genes for sexual characters are present, somatic characters are present on both autosomal chromosome as well as sex chromosome, but the genes for controlling the genes for controlling the sexual characters are present only in the sex chromosomes. Okay, now the chromosomes can further be classified into three groups: homologous, non-homologous, and homologous. So suppose these are two chromosomes. Okay, suppose this is chromosome number 5, this is chromosome number 5. Okay, so these are two chromosomes, and these two chromosomes will have same number of genes, same sequence of genes, and same type of genes. So if the two chromosomes have same number of genes, same types of genes, and same sequence of genes, that those two chromosomes will be called as homologous. Let me explain you with the help of diagram then. So these are the different locus. Okay, suppose here I am having capital A, small a, here I am having small a, Okay, here suppose I am having capital B, capital B. Okay, and here suppose I am having small d, and suppose here I am having capital A, small a. Okay, so in this case, how many genes are present? I will say 1, 2, 3, 4. Why not 5? Because A and A, they are same genes. So how many genes are present? 4. How many types of genes are present? I will say 4 types of genes are present. A, B, D, E. What is the sequence of genes? A, B, D, E. This is the sequence of genes. So you can see both these chromosomes, they have same types of genes. Okay, same sequence of genes and same number of genes. Here I am having A, B, D, E. Here also A, B, D, E is present. Here the sequence of gene is A, B, D, E. Here also A, B, D, E. You do not see. Here you may say, sir, the sequence here is capital A, small a. You do not have to worry about that, because capital A and small a, they belong to the same gene. So here the gene is A, not capital A, small a. Here the gene is A. Here also the gene is A. In both of these cases, the gene is same. Allele is different, but gene is same. Okay, so I will say in both these cases, the sequence of genes, the number of genes, and the types of gene is same, and such chromosomes are said to be homologous chromosomes. And during the process of synapsis or the meiosis or Purkinje phase, they undergo synapsis or pairing. So which chromosomes they pair during synapsis? It is the homologous chromosomes. So when do you call two chromosomes as homologous? If they have same number of genes, same types of genes, and same sequence of genes. Okay, but if the two chromosomes, they do not have same number of genes, same types of genes, and same sequence of genes, they will be called as non-homologous chromosomes. Suppose, let me take an example of chromosome 5 and chromosome 6. Okay, so chromosome 5 homologous, chromosome 6 homologous, chromosome 1 homologous, chromosome 7 also homologous. So but if I say 5 6 or 7 8 or 5 2 or 5 9, they are non-homologous. So suppose, let me take some genes here. Okay, suppose here I am having A, capital B, and suppose here I am having C, D, E. So these are the different genes, and here I am having different genes. So suppose here I am a P, Q, R, S. So you can see the genes present in 5 and genes present in 6, they are different. The number of genes is also different. The sequence of genes is also different, and those chromosomes are said to be non-homologous. So what do you understand by homologous chromosome? Let me give you one example. Suppose this is chromosome X and suppose this is chromosome Y. Okay, so in this case, suppose I am taking two genes here. Suppose the gene is capital A and small b. Here also the genes are present, small a and suppose small b are present. That means you can see that there is similarity. That means in this region, these regions are present in both chromosome X as well as chromosome Y. Okay, chromosome X as well as chromosome Y. And suppose, let me take some sequence over here. So suppose here I'm having PQ and here also I'm having PQ. That means this sequence is present in both chromosome X as well as chromosome Y. Okay, but suppose in this case, let me take some sequence here. Suppose let me take MNO, and suppose here let me take some sequence like STV. Okay, in this case you see the genes are exactly same. Here AABDE, here also AABDE, but in this case some similarities are there. Okay, in this region similarity is there, in this region similarity is there, but in this region there is some differences. In this region there is some differences. Here there is no similarity. When there is no similarity, non-homologous. When there is complete similarity, homologous. When there is some similarities, like in this region, in this region, in this region, in this region, but there is differences also in this region, then those chromosomes will be called as homologous. They will be called as homologous chromosome. What do you call them? You call them as homologous chromosome. Okay, and this region is called as differential region of chromosome X. This region will be called as differential region of chromosome Y. Okay, this region is called as pseudo-autosomal region. This region is called as pseudo-autosomal region. This is called as pseudo-autosomal region. This is called as pseudo-autosomal region. That is what I'm going to write here now. This is pseudo autosomal region 1. This is pseudo autosomal region 1. This is pseudo autosomal region 2. This is pseudo autosomal region 2. This is differential region of chromosome X. This is differential region of chromosome Y. Differential region of chromosome Y. Okay, that means the genes present in the differential region of chromosome Y, they will not be present in chromosome X, and the genes present on the differential region of chromosome X will not be present on chromosome Y. But the genes present on pseudo autosomal region, they will be present in both X as well as Y. Okay, and these chromosomes also pair during synapses. So which type of chromosome synapse during meiosis? Both homologous as well as homologous. Which do not pair during synapses? It is the non-homologous chromosome. Chromosome 1 homologous, chromosome 5 homologous, chromosome 70 homologous. But when I say chromosome 5 6 or 1 2 or 3 4 or 3 9 or 3x or 3y, they are non-homologous chromosomes. Let me repeat. If the two genes have same types and sequence and number of genes, they are homologous. If they do not have same types of genes, they will be non-homologous. If they have some genes common, they will be called as homologous chromosomes. Okay, now let me tell you about phenotype. What do you understand by phenotype? Phenotype is any feature, any distinct feature of organism which can either be observed or measured. Those observable or measurable distinct feature of organisms are called as phenotype. For example, when I say tall, when I look at a pea plant, I can see, okay, this is tall pea plant. Okay, this one is dwarf pea plant. Okay, this person is white, this person is black. So I can observe them. This person is fat, this person is thin. I can weigh them, I can observe them, I can measure them, I can observe them. So those observable or measurable feature of organisms are called as phenotype. I have written some examples here. Okay, so what do you understand by genotype then? See, one individual will have one trait of a character. It will have many traits, but for one character it will have one trait only, right? So for that trait or for that character, what will be the genotype? What will be the genetic makeup, or what will be the genetic or allelic composition of that individual for that trait or character? That allelic composition or genetic makeup is called as genotype. So let me give you one example. So when I say tall plant, so this is phenotype. Okay, and for this tall trait, the individual may have the allele capital T, or it may have the allele like capital T small t. So in this case, capital T or capital T small t, they are the genotypes. This is the phenotype. Or when I say dwarf, this is a phenotype, and for this trait the individual will have two alleles, and the two alleles will be small t. So here these are the genotypes and these are the phenotypes. Okay, that is what I have written over here. And one more thing, the genotype can be homozygous, can be heterozygous, or can be hemizygous. Okay, and the homozygous are also called as pure line. So whenever I say capital T, this means pure line. Whenever I say small t, this means pure line. Whenever I say capital A, this means pure line. Whenever I say small a, pure line. Small b, pure line. Okay, so whenever we say homozygous condition, that will be pure line or true breeding. The pure line can be of two types: dominant pure line or recessive pure line. So in this case, this is dominant pure line, this is dominant pure line, this is recessive pure line, this is recessive pure line, this is also recessive pure line. And the heterozygous condition is called as hybrid condition. Okay, homozygous is called as pure line or true breeding, heterozygous is called as hybrid condition. Okay, now so here I can say, suppose these are two homologous chromosomes. So let's say this is chromosome number 5, this is chromosome number 5. So these white dots are basically locus. we know every locus will have one allele. So here suppose I'm having, in this locus I'm having capital A, here small b, here capital D, here also capital A, small b, small d. Okay, so you can see here, one locus is having one gene and the alleles are of same type. And in this case also I'm taking one locus and the alleles are of same type, but in this case one locus I'm taking but the alleles are of different types. And this condition in which the alleles are of same type in the same locus, that is called as homozygous condition. What do you call that condition? Homozygous condition. This here also the alleles are of same type, so I will say this is homozygous condition or pure line condition. This is dominant homozygous condition and this one is recessive homozygous condition, and this one is called as heterozygous condition or hybrid condition. Okay, so this is these two are homologous chromosomes. So what about the hemizygous condition? So suppose this is chromosome X and this is chromosome Y of which organism? Drosophila. So in case of Drosophila, the genes responsible for controlling the eye color and body color, they are present in the X chromosome, on the X chromosome. So W or W plus, they are going to control the eye color, and Y or Y plus, they are going to control the body color. Okay, so in this case you can see the gene W or W plus is not present on the Y chromosome. Only once it is present. In this case, only once it is present, and this condition is called as hemizygous condition. And here also the Y is present only once, and this is also called as hemizygous condition. And W plus is dominant, so this is dominant hemizygous, and this is recessive, so this is recessive hemizygous. This is dominant hemizygous and this is recessive hemizygous. Recessive hemizygous. Okay, when the recessive allele is present, allele is present in hemizygous condition, it will express itself. But if the recessive allele is present in heterozygous condition, it will not express itself. In this case, the small d will not express itself, but in this case, this small y will express itself. This is the beauty. And in this case, capital D expresses itself. Here also the capital W plus will express itself. Okay. So when I say dominant allele, the dominant allele will express itself in both homozygous dominant condition or heterozygous condition, or even in the hemizygous condition. But when I talk about the recessive allele, it expresses itself only in the homozygous condition and the hemizygous condition. Okay. That is what I am going to write here. And before that, let me give you some more examples. So when you say XY, we know the gene responsible for hemophilia is present on the X chromosome. And if the gene is present in dominant form, it will not cause hemophilia. But if the genes are present in the recessive form, that will cause the hemophilia. Okay. So suppose capital H is the dominant allele. So in that case, it will not be hemophilic. Okay. XY means the person will be male, so this will be normal male. This will be normal male. And here I am writing small h, that means the allele responsible for hemophilia is present in recessive form, so this will be XHY, and this person will be hemophilic male. And here XX, XX, that means these are the females. Okay, when I write smallest, that means I can write here XHXS. Okay, and this lady will be hemophilic lady. Okay, here capital H and small h, that means I can write here XH. Okay, and this lady will be carrier, normal but carrier. And here HH, capital HH, but that means I can write here XX. That means the gene is present in dominant form. Okay, so you can see here, this is heterozygous condition. This is heterozygous condition, this is homozygous condition, and this one is hemizygous condition. This one is hemizygous. This is dominant hemizygous condition. This is recessive hemizygous condition. Okay, this is homozygous condition, this is heterozygous condition. So in this case, small h is not expressing itself, and that is why this lady is not hemophilic. Why? Because capital H is present, but here only one H is present. That means this is hemizygous condition, and in this case, the H is expressing itself. So when the recessive allele expresses itself, if the genotype is either hemizygous like here or homozygous like here, only on those cases the recessive allele will express themselves. Okay, now let me tell you something more about the dominant allele and recessive allele. The allele which express itself in both homozygous and heterozygous condition, they are called as dominant allele. So let me tell you one thing, whether an allele is dominant or recessive, to determine that you have to look at the heterozygous condition. Why? Let me tell you. So when I write capital T or small t, this is tall and this is dwarf, and we know capital T is responsible for tallness, small t is responsible for dwarfness. So in this case, capital T is expressing itself. In this case, small t is expressing itself. But when I write capital T, small t, the phenotype is tall. That means in this case, capital T is expressing, but small t is not expressing. Okay, so in this case, capital T will be dominant and small t will be recessive. So those alleles which express themselves in heterozygous condition, they will be dominant. Which do not express themselves in heterozygous condition but express themselves in homozygous condition only, they will be recessive. That is what I have written here. And for representing them, we use different symbols for these dominant and recessive allele. For dominant allele, we use the capital alphabet like capital T. For example, the dominant phenotype is tall, so I will take the first alphabet and I will write the capital alphabet. First letter is T, and I will take the capital alphabet. Okay, or I can take the W plus. That means, for example, when I say eye color in case of Drosophila, it is white or yellow, white or red. White is recessive, red is dominant. So in this case, I will take the first alphabet of the recessive one, and the first alphabet is W. So if I read only W, that means this is recessive, and if I read W plus, that will be dominant, or I can write plus. Okay. So for recessive allele, I will take the small alphabet of the dominant phenotype, or only the small alphabet of the recessive phenotype, or minus. So these are the different ways to mention or express the dominant and recessive alleles. Okay, now phenotype and genotype. So if I take the pea plant, tall is the phenotype and dwarf is also another phenotype. So the genotype for tall pea plant can be capital T or can be capital T, small t. For dwarf plant, it will be small t. That means I can conclude here that one phenotype is having two genotypes. One phenotype is having only one phenotype. That means one phenotype may have one or more than one genotypes. One or more than one genotypes. Okay. Now, monohybrid cross. So whenever we perform a cross between two organisms, those organisms will have a number of genes. Okay, and if the two individuals, they differ from each other only in one gene, but they resemble with each other in other genes, that cross will be called as monohybrid cross. Let me repeat once again. Whenever I perform any cross, then all the genes will be present in the parent 1 and parent 2. All the genes will be present, but these two parents will be differing in only one gene pair, and that cross will be monohybrid cross. If the two parents which are used in the cross, they differ in two gene pair, that will be called as dihybrid cross. If they differ in three gene pair, that will be called as trihybrid cross. Okay, so the cross in which two parents differ in one gene or one gene pair, that is called as monohybrid cross. And when Mendel performed monohybrid cross, from the results or from the observations of his monohybrid cross, he proposed two laws. One was law of dominance and second was law of segregation. Okay, so one more thing you have to remember, if I write P1, P2, this means this is parent 1 and this is parent 2. So when I perform crossing, when I perform crossing or pollination between parent 1 and parent 2, I have to perform cross pollination. I have to perform cross pollination, and I will get the F1. From F1 I will get F2, from F2 I will get F3, and from F3 I will get F4, and from F4 I will get F5, so on. So the F1 is always obtained by cross pollination, and all the remaining upcoming or further generations, they are obtained by selfing. This is important. F1 is always obtained by cross pollination, whereas the remaining progenies or generations, they are obtained by self pollination. Okay, and as I have said, what is monohybrid cross? The cross in which two parents differ in only one gene. In other genes they will be similar, they will resemble with each other. That will be called as monohybrid cross. So in this question, which one of the following is monohybrid cross? See, in this case, these two parents, this is one parent, this is another parent, they are differing from each other. So this is monohybrid cross. Yes, this is monohybrid cross. What about this one? Here capital A, here also capital A. Here capital D small d, here also capital D small d. So they are resembling, both these parents are resembling in these two genes, but they are differing in, here you can see capital B small b, and here capital, so sorry, here small b, here capital B. So that means these two parents, they differ in only one gene. That is why this will be also monohybrid cross. This will be also monohybrid cross. But in this case, see, here in the third case, here I have capital A, here also capital A. Here I am having small b, here capital B. That means in one gene, these two parents are differing. Here I am having small d, here I am having capital D. That means this is an example of dihybrid cross, because these two parents, they differ from each other in two genes, in gene B and in gene D. That is why this is dihybrid cross. So the answer will be more than one. Okay, so this is an example of what? This is an example of monohybrid cross. This is also an example of monohybrid cross, and this is an example of dihybrid cross. Okay, dihybrid cross. You do not have to see, most of the students, they think, okay, this is not monohybrid cross. Why? Because there are three genes. No, you have to remember, all the genes will be present, but the two parents will differ from each other only in one gene pair. That will be monohybrid cross. Okay, and remember one thing, these parental generations, they have to be always pure line. They have to be always pure line. Okay, that means one will be dominant homozygous, another will be recessive homozygous. Okay, so if I say capital D small t and capital D small t, this is not monohybrid cross. Okay, or if I say small t and small t, this is not monohybrid cross, because one has to be dominant and one has to be recessive. That means this will be this is dominant homozygous, this is recessive homozygous. So this is the monohybrid cross. This is not monohybrid cross, this is not monohybrid cross, this is the monohybrid cross. Okay, now let me explain you monohybrid cross in more detail. So suppose here P for parental generation. As I have told you that the parental generation must always be pure line. One has to be dominant pure line, another has to be recessive pure line. Okay, so suppose, let me take one plant, and that plant is suppose capital T. The genotype is capital T. So the phenotype will be obviously tall. It will be tall. And the second parent is small t. The second parent is small t. So this one will be tall, and this one will be dwarf. This one will be dwarf. Okay, this is parent 1, this is parent 2. Okay, this is dominant parent, this is recessive parent. This is dominant parent and this is recessive parent. Okay, and these parents will produce gametes. They will produce gamete. So here 4 gametes will be produced. from one cell four gametes will be produced, and but only one type of gamete will be produced. Okay, so the gamete will be having capital T, and here the gamete will be having small t, and these gametes will fuse with each other, and after fusion they will produce first filial generation, and that will be having capital T, small t. Okay, so this is heterozygous condition, and the heterozygous condition is called as hybrid condition. So how many heterozygous condition is present? One. So I will call this as monohybrid condition. This is called as monohybrid condition, and this phenotype will be tall. The phenotype will be tall. Okay, now just a minute. This one is F1 generation, and the F1 generation produces F2 generation by self-pollination, by selfing. So to produce F2 generation, I have to perform selfing of F1. I have to perform selfing of F1. So when I say selfing of F1, that means I have to take the same genotype twice. Okay, so this is selfing of what? F1 generation. So it will produce two types of gametes. 4 gametes will be produced, but 2 types will, and here also 2 types of gametes will be produced. Okay, so let me write those gametes here. So these are the F1. These are the gametes of parental generation. These are the gametes of what? Now I'm going to produce the gametes of F1 generation. Okay, so F1 gametes. Now I'm going to produce the F1 gametes. So from here I will get 2 gametes. Here also I'm going to get the 2 types of gametes. The gamete will be capital T. Here the gamete will be small t. Here capital T, and here small t. And these gametes will fuse randomly. This gamete may fuse with this one, and this gamete may fuse with this one. This may fuse with this one, and this may fuse with this one. So here the genotype will be capital T. Here the genotype will be capital T, small t. Here the genotype will be capital T, small t, and here the genotype will be small t. And these are the F2 generation. These are the F2 generation. These are F2 generation. Okay, so in F2 generation, in case of F2 generation, so I can write here in F2 generation. In which cross? In monohybrid cross. In monohybrid cross, in F2 generation, what is the genotypic ratio? The genotypic ratio is 1:2:1. Why? Because capital T is present once, capital T small t is present twice, and capital small t is present once. So the genotypic ratio will be 1:2:1. So I can write here, this will be for capital T, this will be for capital T small t, and this will be for small t. What about the phenotypic ratio? What will be the phenotypic ratio? This is tall, this is also tall, this is tall, and this one is dwarf. That means in F2 generation, 1 is dwarf and 3 are tall. So I can say the phenotypic ratio is 3:1. 3:1. So here this 3 is indicating the tall plants, that means dominant phenotype, and 1 is indicating the dwarf plants, that means recessive phenotype. So in case of monohybrid cross, in F2 generation, genotypic ratio is 1:1 and phenotypic ratio is 3:1. This is dominant, this is recessive. Okay, this is homozygous. This is homozygous. This is dominant homozygous. This is recessive homozygous, and this is the heterozygous. This is the heterozygous or hybrid condition. So this is monohybrid cross. This one is monohybrid cross. So let me write here. This is monohybrid cross. Okay, now what about the dihybrid cross? So when two individuals are crossed, and if the two individuals they resemble with each other in all genes except 2 genes, then that cross will be called as dihybrid cross. Okay, and when Mendel performed dihybrid cross in pea plant, he obtained from the observations of of his results, he proposed the law of independent assortment. From monohybrid cross, he gave law of dominance, law of segregation, and from dihybrid cross, he gave the law of independent assortment. Okay, so in case of dihybrid cross, how the dihybrid cross can be performed, let me explain you here. So here I am going to explain the dihybrid cross. Which cross? Dihybrid cross. Okay, and before that, let me tell you one thing. In case of pea plant, the seed shape can be of two types. The seed shape is controlled by one gene, and it has two alleles. One is capital R and so another is small r. Okay, so the seed shape can be round or can be wrinkled. Can be round or can be wrinkled. Okay, here I have mentioned here. You can see that. Here you can see that. Okay, seed shape. Spherical means round, and this one is wrinkled. Okay, wrinkled. Now you can see seed color. This is yellow and this one is green. Okay, for yellow I will write capital Y. For green I will write small y. Okay, that is what I am going to use here. Okay, and the seed shape and this seed color can be yellow or can be green, and in this case the round seed is dominant, and this is it is due to the dominant allele capital R, and this is due to the recessive allele small r, and this is due to the dominant allele Y, and this is due to the recessive allele Y. Okay, so whenever capital R is present, the phenotype will be round. Whenever the small r is present, when the small r is present twice, that is if it is in homozygous condition, it will be round. Whenever capital Y is present, it will be yellow. If small y is present twice, then only it will be green. Okay, so now let us talk about the dihybrid cross. Which cross? Dihybrid cross. Okay, now so in this case, just a minute. So in this case, P means parental generation. So let me write here. One parent is having capital R, small capital Y. Okay, let me ask you one question. Can I call this one as dihybrid cross? Of course I can call this as dihybrid cross. Okay, this was homozygous recessive, this is homozygous dominant. So this is one monohybrid cross. This is dominant, this is recessive, this is another monohybrid cross. So this means two monohybrid cross means dihybrid cross. Two monohybrid cross means dihybrid cross. Okay, so this one is dihybrid cross, and if I write capital R, capital Y, small r y, and small r capital small y, this is also dihybrid cross. Both are dihybrid crosses. Both are dihybrid crosses. Okay, now so one parent is having this genotype and the other parent is having small r, small y. This is the genotype of other parent. Okay, so I'm performing cross pollination here. Which pollination? Cross pollination here. So what will be the phenotype here? Capital R is present, that means round seed will be there, and the seed color will be yellow because capital Y is present. So round yellow seeded plant, and here wrinkled green seeded plant. They were crossed with each other. Okay, so this plant will produce the gamete and this plant will also produce the gamete. So it will produce one type of gamete. Here I am talking about the types of gamete. So it is going to produce one type of gamete, and the gamete will be capital R having capital R with small y, capital Y, and this will have the small r with small y, and these gametes will fuse with each other. They will fuse with each other, and after fusion, that means they will fertilize, and after fertilization they will produce the F1 generation. That will produce the F1 generation, and the F1 generation will be capital R, capital Y, small y. Okay, so you can see Heterozygous condition is dihybrid. So this is one hybrid, this is another hybrid, that means this is dihybrid. This is dihybrid. So I can write here, this is an example of what? Dihybrid. This is an example of dihybrid. Okay, and the phenotype will be, round yellow seated. Which one? Round yellow seated. And now I will perform the selfing of F1, and after selfing of F1 I will get the F2 generation. Okay, so I can say the selfing of F1 will produce the F2 generation. Okay, so how the F2 generation will be produced, let me explain you here. How the F2 generation will be produced, let me explain here. Okay, you do not have to draw the Punnett square, it is not necessary. I'm giving you one trick here, and that trick will be useful to you. Okay, so here I'm writing the F2 generation. So first I will write the genotype, then I will write the phenotype. F2 generation of dihybrid cross. Okay, so you please write first here capital R 3 times. Okay, you have to write this one 3 times. Then you please write capital R small r 3 times. Then write small r 3 times. So this one 3 times, this one 3 times, and this one 3 times. Now you write capital Y, then capital Y, and small y. This pattern you have to follow here also. Okay, that means I have to write here capital Y, capital Y, small y. Here capital Y, capital Y, small y. So all these are the 9 genotypes which we will get after in the F2 generation in dihybrid cross. In the dihybrid cross, if the genes are unlinked. Okay, so the individual having this genotype and this genotype, do you think they will be produced in same number? No, they will be produced in different numbers. So what is the number of the individuals with this genotype in F2 generation? What is the number of individuals of this genotype, of individuals with this genotype? To remember this, there is a trick, and that trick also I am going to write here. You have to remember one thing, 1 2 1 2. So you have to just follow that one. You please write 1 2 1 2 1 2 1 2 1. Okay, and this one is dihybrid, this is one hybrid, this is another hybrid, so this is dihybrid, and only this is the only dihybrid case. Otherwise, in other cases, either homozygous or monohybrid case is present, but this is the only case where dihybrid condition is present. Okay, and this one, for this one you have to write this one as 4. That means this sequence, the ratio will be 1 to 1 to 1 to 1 to 4 to 1 to 1. 1 to 1 to 4 to 1 to 1. Very simple. Now what will be the phenotype then? The phenotype will be very easy. Okay, so capital R is there, that means this will be round, and capital Y is present, that means this will be yellow. This is also round yellow. This is going to be round and green. Okay, this is going to be round and yellow. Okay, this one is also round and yellow. This is round and green, right? And this is wrinkled and yellow. This one is also wrinkled and yellow, and this one is wrinkled green. Wrinkled and green, right? So the genotypic ratio is 1 to 1 to 4 to 1 to 1. So what about the phenotypic ratio? The phenotypic ratio, the 4 different types of phenotype are produced. So I will write here in F2 generation of dihybrid cross. Which cross? Dihybrid cross. The genotypic ratio. Which ratio? Genotypic ratio is 1 2 1 2 4 2 1 2 1. What about the phenotypic ratio? What about the phenotypic ratio? So four different types of phenotypes are produced. What are those? One is round yellow. Okay, round is dominant phenotype, yellow is also dominant phenotype, so I can write here this is dominant. So for dominant I'm writing D. Okay, and the second one will be round green. Round is dominant, green is recessive, so this is dominant recessive. The third one will be wrinkled yellow. Okay, fourth one will be wrinkled green. Okay, and this will be recessive dominant, and this is recessive. Okay, so this pair round yellow, you can see round yellow is the phenotype of what? Parent, dominant parent. Wrinkled green is the phenotype of recessive parent. So wrinkled green and round yellow, they are the parental phenotype. Okay, so that is why I can say these are this one and this one, they are the parental phenotype, and these two are the recombinant. So I will write here, this is parental phenotype, this one is parental phenotype, and these two are non-parental phenotype, or you can say recombinant phenotype. So how many round yellow you are going to get? If you calculate here, so you are going to get how many round? 9 round you are going to get. 3 round yellow you are going to get 9. Round green you are going to get 3. Wrinkled yellow you are going to 3, and wrinkled green you are going to get 1. So the ratio will be 9:3:1 ratio. The ratio will be 9:3:3:1. This is the ratio. Okay, and in this case, this is the parental genotype, and all these remaining are the non-parental or recombinant genotype. Okay, that means this is parental, and all the remaining are the recombinant. Okay, that is what I'm going to write here. Okay, so this one is parental, P for parental, and all the remaining are the recombinant. So these are the recombinant. So what is the total recombinant produced? It is the 14 out of 16. What is the total parental produced? 2 out of 16. Okay, how many dominant parental are produced? 1 out of 16. How many recessive parental genotypes are produced? 1 out of 16. Okay, so this is about genotypic ratio. Okay, so this. Now let me tell you more about the crosses. So let me tell something about back cross. So what do you mean by back cross? If the F1 generation is crossed with any of the parent, that is called as back cross. So if F1 is crossed with dominant parent, that is called as out cross. And if the F1 is crossed with recessive parent, that is called as test cross. Okay, so this is F1 and this is the parent. This is dominant parent. So this is the out cross. This one is F1 and this one is recessive parent. So this is called as test cross. Okay, so the back cross is of two types, out cross and test cross. If the F1, that is capital D small t, is crossed with either with the dominant parent or with the recessive parent, they are called as back cross. If recessive parent is taken, test cross. If dominant parent is taken, that is called as out cross. Okay, that is what I have written here. Okay, so in this case, what will be happening here? So I will get capital T and I will get capital D small t. I will get this one 1 and this one also 1. So one tall, this one also tall. So that means 100% tall I will get. In this case, I will get two types of genotype. One will be capital D small t, another will be small t. This is tall and this is dwarf. So in case of this is one and this is also one. So in case of test cross, monohybrid, this is monohybrid test cross. Okay, this is outcross. So in case of monohybrid test cross, what is the ratio? Genotypic ratio 1:1. What is the phenotypic ratio? 1:1. Okay, and this is dominant parent, this is recessive parent, and this one is F1. Okay, so if this F1 is crossed with this one, that means this, that will be outcross. If this F1 is crossed with this one, that means recessive parent, that means this one, that will be called as test cross. Very easy, right? So what is the significance? What is the significance of the test cross? What is the significance of test cross? The test cross is done to check whether the genotype of the dominant phenotype is homozygous or recessive, or homozygous or heterozygous. Okay, so for example, if I say tall plant, this is the plant with dominant phenotype. So by looking at the phenotype, can you say that the genotype will be capital D or capital D small t? No, you cannot say that. The plant with this dominant phenotype may have this genotype or may have this genotype. This is homozygous, this is heterozygous. So how to check the genotype of this dominant phenotype? To check that, I will perform the test cross. Okay, and if after test cross only dominant phenotype are produced, then that plant will be that tall plant will be small capital D, and if both dominant and recessive phenotype both are produced, that means that will be heterozygous. Okay, and this test cross is also done to find out the types of gametes produced by an individual with a given genotype. That means how many types of gametes are produced by an individual. To calculate that, you have to cross that individual with recessive parent. That means you have to perform test cross. By doing that, you can calculate the number of gametes produced by that individual. We can also use this test cross to find out whether the genes we considering in a cross are linked or unlinked. Okay, for example, if I take dihybrid cross, let me write the ratio first here. Okay, test cross ratio. So which ratio I am taking here? Phenotypic and genotypic ratio, both ratio I am taking here. So in case of monohybrid cross, what is the test cross ratio? It is 1:1. In case of dihybrid cross, it is 1:1:1:1. When if the genes are unlinked. But if the genes are, if in case of dihybrid test cross, if the genes are linked, then the ratio will be 1:1. Okay, so in case of dihybrid test cross, the ratio can be 1:1:1 or can be 1:1. Depends on the nature of genes. If the genes are unlinked, the ratio will be 1:1:1:1. If they are linked, it will be 1:1. Clear? Like that, in case of trihybrid cross, if the genes are unlinked, the ratio will be 8:1. And if the genes are linked, then the ratio will be 1:1. So whatever number of genes you take, if the genes are linked, then the ratio will be 1:1. And remember one thing, one dihybrid cross means 2 monohybrid cross. One trihybrid cross, that means that will be 3 monohybrid cross. 3 monohybrid cross. Clear? Now, so when the great Mendel performed his experiment on pea plant, he took 7 characters. How many varieties or traits he considered? He considered 14 varieties or 14 traits. Okay, and he considered 7 characters. So what are those characters? Here it is written: seed shape, seed color, flower color, pod shape, pod color, okay, flower position and stem height. These are the different characters. 7 characters he took, and he performed his experiment, hybridization experiment on pea plant for 7 years, from 1856 to 1863. Okay, the duration was 1856 to 1863. He performed his experiment, and his experiment was hybridization experiment on pea plant. On which plant? Pea plant. Okay, and in case of pea plant, the pea plant is diploid, and what is the value of diploid? 14. That means in this case 7 different types of chromosomes are present. Okay, so pea plant who took for his experiment, he performed his experiment for 7 years. Okay, he considered 7 characters. He considered 7 characters. Okay, and these are the characters, and you can see the dominant phenotypes are here and the recessive phenotypes are also present here. Okay, now let me tell you about the law of Mendel's law of inheritance. Okay, but before that, let me drink some water and I will start again. So now let's talk about the laws of inheritance. So from the observations of Mendel's experiment, he gave some conclusions, and those conclusions were later on given the name of laws of inheritance. So the first law of Mendel was law of dominance, and the second law was law of segregation, and the third law was law of independent assortment. So what does this law of dominance, or the first law of Mendel, says? Okay, so before that, let me repeat one point once again, that this first law of the first law of Mendel, that is the law of dominance, was observed from the result or the observations obtained from the results of the observations of what? Monohybrid cross. Okay, and this is the first law of Mendel, and this is not universal, and this law is not universal. That means this law has some limitations. Why? Because this is not applicable in for all characters. For all characters, this law is not applicable, and that is why this is not universal. So what are the exceptions to this law? One is incomplete dominance, second is the codominance. Okay, and this law of dominance states that for every character there are factors. That means every character is controlled by factors, and every character and the factors which are responsible for controlling a character, they always occur in paired condition. I repeat, every character is controlled by factors, and those factors for each character are present in every individual in paired condition. And in paired condition, the factors may be of similar type, and that condition will be called as homozygous, or may be of dissimilar type, and that condition will be heterozygous. And in heterozygous condition or in dissimilar condition of paired factors, only one factor will be able to express itself, and that factor which can express itself in heterozygous condition is called as dominant factor, while the other factor will be called as recessive factor. Okay, let me repeat. From the observations of Mendel's monohybrid cross, he gave this law, and this is the first law of Mendel. Which law? Law of dominance. And this has some exceptions. Because it has some exceptions, like incomplete dominance and codominance, that is why this is not universal. And this law says that every character is controlled by factors, and those factors in every individual, they occur in paired condition, and in that paired condition, those factors can be of similar type, for example capital T or small t. That condition will be called as homozygous, or they can be different types, for example capital T small t, that will be called as heterozygous. In that heterozygous condition, only one factor will be able to express itself. That factor is called as dominant factor, the other factor will be called as recessive factor. Okay, and because of so, and you can see capital T is responsible for tallness, small t is responsible for dwarfness, and the genotype capital T small t is responsible for producing the phenotype tall, dominant phenotype. That means here the capital T is expressing itself, whereas small t is not expressing. That is why capital T is dominant, small t is recessive. Okay, fine. And in case of monohybrid cross, the phenotypic ratio, F2 phenotypic ratio is 3:1, and this is due to law of dominance. This is due to law of dominance. Why? Because you know very well that in monohybrid cross, in F2 generation, the phenotypic ratio is 3:1. 3 tall are produced. This is tall, no problem. But these two are also tall. And because these two are tall, that is why the tall plant are 3 in number. Tall, 3 in number. And this happened because in these two cases, capital T is expressing, but small t is not expressing. That means dominance is observed here. So I can say because of dominance, this 3:1 ratio is obtained. 3 tall and 1 dwarf is obtained. Okay, so you can write that point there in law of dominance, that 3:1 phenotypic ratio in F2 generation in monohybrid cross is due to law of dominance. Law of dominance. Clear? Let me write that part here. In case of monohybrid cross, which cross? Monohybrid cross. Three is to 1 phenotypic ratio is due to dominance. It is due to dominance. Okay, now so let's compare the dominance with incomplete dominance and codominance. Okay, so I have taken the character here. The character is height of tree plant. Okay, so this is parental generation. So capital T, small t, this is dominant parent, this is recessive parent. Okay, so this individual will be tall. Okay, and this individual will be dwarf. And when we cross them, I will get capital T, small t, and this is also tall. This is also tall, and this is F1 generation. So you can see this F1 generation is resembling with this one with respect to phenotype, not with respect to genotype. Okay, so I will say with respect to genotype, phenotype, with respect to phenotype, this F1 tall is resembling with one of the parent. These are the two parents. One parent is dwarf, one parent is tall, and this F1 is tall. That means I can say this F1 resembles phenotypically with one parent. Which parent? Dominant parent. Which parent? Dominant parent. So I can say in case of dominance, the F1 resembles with either parent. Okay, and in this case you see the F1 is having capital T, small t, and the phenotype is tall. That means in this case capital T is expressing itself 100% and small t is expressing itself 0%. That means here one is expressed completely, another is expressed 0. That means I can say capital T is dominating the expression of small t, and this is called as dominance. Now if you perform the selfing, of F1, you will get the F2 generation. Okay, you will get this one 1, this one 2, and this one 1. This will be tall, this is also tall, and this is also this is dwarf. Okay, this is dwarf. So in F2 generation, what is the genotypic ratio in case of dominance? The genotypic ratio is 1:2. See, 1:2:1. So the genotypic ratio is 1:2:1. What is the phenotypic ratio? 3 tall, 1 dwarf. So the phenotypic ratio is 3:1. Where? In case of dominance. And whenever you say dominance, you have to think with respect to complete dominance. Which dominance? Complete dominance. Now what about incomplete dominance? Let me explain you with the help of example of Mirabilis jalapa. So in case of Mirabilis jalapa, which is also known as 4 o'clock plant, the flower color is controlled by one gene, and that gene has two alternative forms, two alleles. One is capital R, second is this small r. So this is parental generation, and the capital R is responsible for red flower, small r is responsible for white flower. So this one will be red, and this one will be white. Clear? And when we cross them, I will get the F1 generation, and the F1 generation is not red. This is pink in nature. This is pink in nature. Okay. So in this case, you can see the phenotype of F1 is pink, and the none of the parent is pink in nature. So I can conclude that in case of incomplete dominance, the F1 generation resembles with neither parent. It does not resemble with dominant parent. It does not resemble with the recessive parents. That is why this resembles with neither parent. In case of dominance, this resembles with either parent or one parent. Which parent? Dominant parent. Okay. Now, in this case, capital R is expressing itself but not 100%. Here, capital T was expressing itself 100%. Here, it is expressing itself but not 100%. It is expressing itself more than 50%. And this one is expressing itself less than 50%. Here, small t was not expressing. Here, it is expressing but less. It is not expressing more. So, which one is expressing more? Capital R is expressing more. Okay. So, I can say there is a blending or the mixing of the phenotype here. Okay. Now in case of F2, how do I get this F2 generation? By selfing of the F1, and after selfing of the F1, I will get this genotype 1, this genotype I will get 2, and this genotype I will get 1. This will be red, this will be pink, and this will be white. Okay, that means I can say in F2 generation, in case of F2 generation, what is the genotypic ratio? The genotypic ratio, here it is. What is the genotypic ratio? 1:1. Phenotypic ratio is also 1:1. So I will say 1:1. Okay, and here also 1:1. That means I can say in case of incomplete dominance, genotypic ratio and phenotypic ratio are same. Okay, so I can say here F2 genotypic ratio is equals to phenotypic ratio, and what is that? That is 1:2:1. What about codominance? In case of codominance, to explain that, I will take the example of ABO blood grouping. Okay, remember one thing. If I write capital IAIO, here this one will express itself completely. This one will not express at all, and the blood group will be A. So in this case, IA is completely dominant over IO. So this is an example of dominance. And if I write IB IO, in this case, this one will express 100%, this one will express 0%. So, and the blood group will be B, and the individual, and in this case, I will say there is dominance, complete dominance. But if I write IAIB, so this one will express itself 100%, and this will produce the antigen A. Okay, and this one will express 100%, and this will produce the antigen B, and that is why the blood group will be AB. Okay, and here this one is expressing 100%, this one also 100%, and that kind of interaction is called as codominance. What do you call that? You call that as codominance. Clear? So let me take two individuals. One is having blood group A, another is having blood group B. So here the blood group is A. Here the blood group is A, and here the blood group is B. Okay, and in this case the blood group is AB. Why? Because antigen A and B both are produced. So in this case I will say IA is expressing itself 100%, IB is also expressing itself 100%. So if only one expresses 100% dominance, if both express but one expresses more but less than 50%, another expresses itself and less than 50%, that is incomplete dominance. And if both alleles express themselves completely, that is called as codominance. Here only antigen A is produced, here only antigen B is produced, here antigen A, B both are produced. So I can say here that the F1 generation resembles with both parents, both dominant and recessive. So I can say in case of dominance, F1 resembles with either parent, F1 resembles with either parent. In case of incomplete dominance, F1 resembles with neither parent. And in case of codominance, it resembles with both parents. And after selfing, I will get the F2 generation. And in F2 generation, this will be blood group A, this will be blood group AB, and this will be blood group B. Okay, and I will get individual like this 1, this will be 2, and this will be 1. So I can say here in codominance also, the phenotypic ratio, F2 phenotypic ratio, F2 phenotypic ratio is equals to F2 genotypic ratio. And what is that value? The value is 1:2:1. The value is 1:2:1. The value is 1:2:1. Clear? Now let me give you some more examples of incomplete dominance and codominance. In case of Mirabilis jalapa, the flower color is controlled by one gene that will be having two alleles, capital R, small r. They show incomplete dominance. In case of dog flower, which is also known as antirrhinum, okay, antirrhinum matches, the flower color is also controlled by one gene, and that also shows that one also shows the incomplete dominance. And in Adensulian fowl color, this character is also an example of what? Incomplete dominance. What about the example of codominance? In case of cattle, the coat color is an example of codominance. In case of ABO blood grouping, AB shows codominance, and in case of hemoglobin, HbA and HbS, they are the example of what? Codominance. Okay, let me write this part here. This is important. So if I write IAIO or IBIO or IAIB, okay, so in both of these cases, these are the example of what? This is the example of dominance. This is the example of dominance. And this one is the example of what? Codominance. That means you can say that ABO blood grouping, ABO blood grouping shows what? It shows multiple allelism. Okay, it shows dominance, and it also shows codominance. It shows codominance. Clear? Leave it. Now let me tell you about the second law of Mendel. And the second law of Mendel was law of segregation. Segregation means separation. Okay. So this law of segregation is known as law of purity of gamete. Why? I will tell you in this slide only. Okay. This is the second law of Mendel, and this law was obtained from the observations of the results of monohybrid cross. Okay, and this law has no exception, and because this law has no exception, that is why this is universally applicable. Law of dominance has exceptions, that is why this is that was not universally applicable, but this one is universally applicable. Some teachers will tell you that it has exception like some exceptions like non-disjunction, but that is not an exception. Okay, so what does this law of segregation says then? It says that if an individual has two factors for a character, then those two factors which are present in an individual will not mixed up, they will not get blending, but that means they keep their identity, and when meiosis takes place, during NFS first. During NFS first. And we know that in case of animals or higher plants, the meiosis takes place during gametogenesis. Let me repeat here. What does this law says? This law says that this law says that the two factors, for example, capital T and small t, which are present together in an individual, they will remain together without getting mixed up. That means they will keep their identity. They will not get mixed up. That means if this is, suppose, if this is capital T, okay, capital T, small t, they are basically alleles. Alleles are nothing but the segment of DNA. So suppose this is capital T and suppose this is small t. So it will not happen. Both of them, they will remain together, and any segment of this one will not get transferred here, and any segment of this one will not get transferred over here, and that is why they will keep their identity. Okay, no exchange of any segment will take place, and that is why they will remain, they will keep their identity. Okay, and when gametogenesis takes place, that means when gamete formation takes place, meiosis has to take place. Okay, and during meiosis, in NFS first, the homologous chromosomes, they will separate, and because the homologous chromosomes separate, that is why the alleles will also get separated. Okay, or that means the factors will get separated, and after the process of meiosis, the 4 cells will be produced, and the 4 cells will be gametes, and each gamete will carry or uptake either capital T or small t. Either capital T or small t. Okay, that means every gamete has only one type of allele, either capital T, purely capital T, or purely small t, not a fragment of capital T and fragment of small t, either completely capital T or completely small t. That is why the gamete is always pure with respect to allele, and that is why this law of segregation is called as law of purity of gametes. The gametes are always pure with respect to allele. Okay, so let me repeat here. This law is universal because this does not have any exceptions. Okay, and this law says that the two factors, for example, capital T and small t, when they are present in an individual together, they stay together without getting mixed up, without any blending. And during gametogenesis, meiosis takes place, and in meiosis, in NFS first, they will get separated, and at the end of meiosis, the 4 cells will be produced, and the 4 cells will be gametes, and those gametes will be having either capital T or small t. So that means the gametes will be completely pure with respect to alleles or factors. That is why these gametes are called as pure gametes with respect to allele, and that is why this law is called as law of purity of gametes. Okay, now what about the law of independent assortment? Assortment is nothing but the separation, okay, or arrangement, you can say. This is the third law of Mendel, and this was obtained from the observations of the dihybrid cross, dominance and segregation, monohybrid cross, and law of independent assortment from the dihybrid cross. Okay, and this is not universally applicable. That means this one also has exception, just like the monohybrid law of dominance, and the exception is linkage. Okay, and this law says that the separation of one pair of factors is independent of the separation of other pair of factors. For example, let me take two pairs of factors. This is one pair of factors. This is another pair of factors. So when meiosis or gametogenesis takes place, capital R and small r, they will get separated from each other, and capital Y and small y will also get separated from each other. Okay, and in this case, the separation of capital R and small r is independent of the separation of capital Y and small y. Okay, and this is called as law of independent assortment. Law of independent assortment. And because capital R, small r, separation of capital R and small r is independent of separation of capital Y and small y, that is why 4 different types of gametes will be produced. How many? 4 different types of gametes will be produced. Okay, that is what I have written here. Because the segregation of one pair of factors, here the one pair of factor is capital R, small r, is independent of the segregation of other pair of factors. The other pair of factor is capital Y, small y, is independent. That is why I can say this one will produce 4 types of gametes. And because 4 types of gametes are produced, that is why the F2 phenotypic ratio is 9:3:3:1. So 9:3:3:1 phenotypic ratio is due to what? Independent assortment. Due to independent assortment. 3:1 ratio is due to dominance, law of dominance. 9:3:3:1 ratio is due to law of independent assortment. law of independent assortment. Clear? Now, just a minute. Where is linkage? Okay. Before discuss, now let us talk about the linkage. Now let us talk about linkage. The exception of law of independent assortment is linkage. That means this law of independent assortment is applicable only if the genes controlling the characters are present on separate chromosomes. Then only that law will be applicable. But if they are present on the same chromosome, that will show linkage, and that means that those characters will not show independent assortment. Okay. So what is linkage? So suppose this is a chromosome, and this chromosome has a number of genes. Suppose this is gene A, gene B, gene C, gene D. These are the genes. Okay, and the characters controlled by these genes are, suppose, character 1, character 2, character 3, and character 4. These are the characters. Okay, this is one chromosome. This is one chromosome. So on a chromosome, a number of genes are present, and those genes, these are the genes, these are the genes, and these are the characters. These are the characters. 1, 2, 3, 4 are characters. A, B, C, D are the genes. Okay, so the genes which are present on the same chromosome, they are called as linked genes. So in this case, AB, ABC, ABCD, BC, BD, all these are linked genes. And the characters controlled by linked genes are called as linked characters. Okay, genes present on same chromosome, linked genes. Genes present on the characters controlled by linked genes are linked characters, and the inheritance of or the transmission of linked characters is called as linkage. Okay, let me repeat. The genes present on a same chromosome are linked genes. The characters controlled by them are linked characters, and how they get transmitted from the parents to the progeny, that is called as linkage. Okay, and suppose let me take another chromosome here. Suppose this is another chromosome, and suppose I'm taking some genes like PQR. Okay, and the characters controlled by them are 5, 6, 7. So in this case, gene A and P, they are unlinked. Gene B and P, they are unlinked. Gene P and Q, B and Q, they are unlinked. And the characters 1 and 1, 5, 1, 6 or 3, 4, 3, 6, they are unlinked characters. Genes present on same chromosome, linked genes. Genes present on different chromosomes, they are unlinked genes. Clear? And this concept of linkage was given by T.H. Morgan, who is considered as father of experimental genetics. And he gave this concept of linkage by performing a dihybrid cross, not a dihybrid cross, two types of dihybrid cross in Drosophila, which is also known as fruit fly. Why? Because this grows normally on ripened fruit like banana. Okay, so he performed two types of dihybrid cross. In one cross, he took two characters like eye color and body color of Drosophila. In another cross, he took eye color and wing size. Okay, so in just a minute. Okay, so these so the characters which were taken by T.H. Morgan in his dihybrid cross were eye color, body color, and wing size. Okay, eye color has two different forms. The dominant form is red and recessive form is white. And for red, I will write the allele will be capital, small w plus, and for white it will be simply w. Body color, for body color, the dominant phenotype is brown and it will be written as y plus, and recessive trait is phenotype is yellow, it is written as small y. Wing size can be normal or normal size or miniature. Miniature is recessive, it is written as small m. The allele is written as small n, and the normal size is written as for normal size, the allele is written as m plus. Okay, so these are the two crosses, and you can see here this is the first cross and this is the second cross. Okay, this is the first cross and this is the second cross. And remember here, this type of chromosome is indicating Y chromosome, and if simply straight line is written, this is X chromosome. Okay, whenever a line is written like this, this is representing X chromosome, and if the line is like this, this is representing Y chromosome. Okay, so here you can clearly see these are two X chromosomes, so this is female. Okay, these are the this is one X chromosome, this is Y chromosome, this is male. So that is, this is why this is father and this is mother. Okay, and you can see here YW. That means Y is responsible for yellow and W is responsible for white eye. So yellow body, white eye, female, and here brown body and red eye, male. When they are crossed with each other, the F1 generation is produced. In F1 generation, the daughter will be having this one genotype, and the son will be having this one genotype. Okay, and because here you can see, you just look at your book and study book, you can see there clearly. Here you can see this is Y and this is Y plus, so it will be brown body. Okay, and here you can see W and W plus, that is why it will be red eye. Okay, brown and red eye, and here YW, that means yellow and white eye. Okay, and here you can, and this F1 generation will produce the F2 generation. So this entire one is the F2 generation. Okay, and in F2 generation I will get 8 different types of combinations. Okay, 1 2 3 4 5 6 7 8 different types of combinations. Okay, and this one is wild type, this one is wild type, this one is a wild type, this one is wild type. Okay, sorry, all these are called as parental generation, and all of these are called as this non-parental generation or recombinant generation. Okay, for parental generation you do not have to look at both chromosomes, you have to look every chromosome individually. So you just look at this chromosome, here it is Y plus and W plus. In parental generation, here I am having Y plus and W plus, so this is parental chromosome, right? And this is Y chromosome, so this is parental. In this case Y and W, here you can see Y and W, they are present together, and this is parental, so this is parental, and this is Y chromosome, so this is parental. Okay, parental type. And here you can see Y, and here Y plus and W plus, Y plus W plus is there, so this is parental, this one is parental, so this is parental. Here YW is present, Y and W, they are present in the parental generation, so this is also parental, because both chromosomes are parental, that is why this is parental. Like this, here this is parental, so this is parental. Okay, but in this case Y plus and W, in parental combination, Y is present with W, and Y plus is present with W plus, and here you can see Y plus and W is present, so this is recombinant type, and this is parental chromosome. So if both chromosomes or one chromosome is recombinant type, then this individual will be recombinant. Okay, here also this will be recombinant because this is Y and this is W plus, so this is recombinant. Okay, Y plus and W, so this is also recombinant, and Y and W plus, so this is also recombinant. So how many recombinant do I get? I get 4 types of recombinant. Okay, all of these are recombinant types, and all of these are the parental combinations. Okay, so how many types of recombinants are produced? 4 different types of recombinants are produced in F2 generation. And how many parental combinations are produced? I will say 4 different types of parental combinations are produced. Okay, and what is the total parental combination produced? 98.7%. So this was produced 98.7 upon 4%. This one also is produced 98.7 upon 4%. This one is also produced 98.7 upon 4%. The probability of getting this one is also 98.4 upon 7%. All the parental types are produced equally with equal probability. What about this one? The recombinants are produced 1.3%. That means the 4 different types are produced will be 1.3%. So this one will be 1.3 upon 4%. This one is also 1.3 upon 4%. This one is also 1.3 upon 4%, and this one is also 1.3 upon 4%. That means all parental types are produced equally and all recombinants are produced equally. Okay, and you can see only 1.3% recombinants are produced. Okay, so if only 1.3% recombinants are produced, that means the recombination frequency will be very, very less. Okay, why? Because we know recombination frequency is equals to total number of recombinants upon total progeny into 100. Okay, so so if the recombination frequency is, so I can say if the recombinant produced are very less, that means the recombination frequency will be very less, and the recombination frequency depends upon the distance between the genes. I repeat, recombination frequency is equals to total recombinants upon total progeny into 100. So if the recombinants produced are less, the recombination frequency will be less, and the recombination frequency depends upon the distance between the genes. If the distance is less, recombination frequency will be less, the recombination will be less. So because in this case only 1.3% of recombinants are produced, that is why the recombination frequency will be less, and because the recombination frequency is less, that is why the distance between the genes Y and W is very, very less. So you can see the distance, you can clearly see the distance between Y and D is very, Y and W very less, Y plus and W plus very less. Okay, in the same way in cross B is happening. Okay, so here white eye and miniature, here red eye and normal wing are taken. Okay, so this is F1 generation and this is F2 generation, but in this case the parental is the, and these are the parental types, these are the parental types, and these are the recombinant types. Okay, all the parental types are produced equally with equal probability, all the recombinants are produced with equal probability. Okay, so I will say this recombinant will be produced 37.2 upon 4%. This one will also be produced 37.2 upon 4%, 37.2 upon 4%, and this one will be produced 37.2 upon 4%. Okay, what about the recombinant, sorry, parental type? Total parental type is 62.8%, so this will be 62.8 upon 4%. This one also 62.8 upon 4%, 62.8 upon 4%, 62.8 upon 4%. So you can see here the recombinant produced is 37.2. Here it is only 1.3%. Here 37.2%. So if recombinant produced is more, that means recombination frequency will be more, and if the recombination frequency is more, that means the distance between genes will be more, and that is why you can see the distance is more. Here the distance is this much, here the distance is this much, but here the distance is this much. So you can see the distance between the gene controlling eye color and wing size is more as compared to the distance between body color and eye color. Okay, so this was about what? This was about the linkage. This was about linkage. And so that is what I have written over here. Because more recombinants are produced in second cross, that is why I will say that the distance between gene for eye color and body color is more, sorry, not more, less. It has to be less. Because in the first case total recombinants produced are less, that is why I will say the distance between genes for eye color and body color is less as compared to the distance between the genes for eye color and wing size. And as per T.H. Morgan, linkage is basically physical association of genes on same chromosome. On same chromosome, the different genes are present together, and that association of genes on the same chromosome, physical association of genes on same chromosome, is called as linkage as per T.H. Morgan. But in modern definition, we say linkage is nothing but the transmission of linked characters. Okay, and the formation of the generation of non-parental type gene combination. Non-parental gene combination is called as recombination as per T.H. Morgan. And for non-parental type, I can write another term like recombinant. So whenever I say recombinant, that is nothing but the non-parental type. Okay, now recombination frequency is nothing but the total number of recombinants upon total number of offsprings or progeny into 100. And this recombination frequency is also called as crossover value. COV is crossover value. Okay, and the recombination frequency, the value of recombination frequency depends upon the distance between the genes. If the distance between gene is more, recombination frequency will be more. Okay, and we can say that while constructing the genetic map, we assume that the value of recombination frequency is equals to the value of distance between the genes. Okay, and the units, when we construct a genetic map, chromosomal map, we use different units for the distance between genes. So the units for distance between genes is of two types: map unit and centimorgan. CM is centimorgan. Map unit we use in case of prokaryotic genes, and centimorgan we use in case of eukaryotic genes. Okay, so this was something about the recombination frequency. Now I will talk to you about the sex determination. Which one? Sex determination. But before discussing sex determination, let me tell you something about the just a minute. Let me tell you something about the chromosomal theory of inheritance. Chromosomal theory of inheritance. It was Sutton and Boveri who gave the chromosomal theory of inheritance. Okay, so let me write that part. So the chromosomal theory of inheritance, it was given by, or it was proposed by Sutton and Boveri. Okay, but this was experimentally proved by T.H. Morgan, and he proved this by experimenting on the Drosophila. Okay, so he experimented on Drosophila. For linkage, he experimented on Drosophila, and for chromosomal theory of inheritance also he experimented on Drosophila. Okay, and these two scientists, Sutton and Boveri, they compared the behavior of chromosomes and genes, which are called as nothing but the factors, during gametogenesis. Okay, so I will say they compared Sutton and Boveri, they compared the behavior of genes and chromosomes during meiosis, or I can say gametogenesis. And they found that their behavior is parallel. For example, so what are the postulates of this chromosomal theory of inheritance? You please write. I am dictating that part. The first point of this theory is that both chromosomes and genes, that is the factors, they occur in pair. Okay, the chromosomes as well as the factors or genes, they occur in paired condition. First point. Second point, that the pairing of homologous chromosomes results in the pairing of factors they carry. Okay, for example, suppose this is chromosome number 5, this is chromosome number 5. Okay, so in during synapses, okay, so suppose this is just a minute. Okay, this is chromosome 5, this is chromosome 5. Okay, suppose this one has capital A, this one has small a. So when these two homologous chromosomes they pair during synapses, so bivalent is formed. These are homologous chromosomes. So these homologous chromosomes they pair with each other. So when they pair with each other, the capital A will pair with small a. The capital A will pair with small a. So I will say that the chromosomes are present in a diploid cell. In a diploid cell, both chromosomes and genes they occur in paired condition. First point. Second point, during meiosis, in zygote phase, during synapses, the chromosomes, the homologous chromosomes, they pair with each other. And the pairing of homologous chromosomes results in the pairing of the genes or the factors or alleles they carry. Third point, during anaphase first, these two chromosomes they will separate from each other. So when they separate from each other, the alleles or the factors capital A and small a will also get separated. Okay, so what are the points? First point, in diploid cell, both genes and the chromosomes they occur in paired condition. In synapses, the chromosomes, the homologous chromosomes they pair with each other, which will result in the pairing of the genes or the alleles they carry. Third point, in anaphase first, when the homologous chromosomes separate, at that time the alleles will also get separated. The capital A will get separated from small a. The capital T will get separated from small t. Okay, and when fertilization takes place, the paired condition will be restored for both for the chromosomes as well as the genes. Okay, so all these are the points of what? Chromosomal theory of inheritance. Okay, now let us talk about the sex determination. So when we look around us, we find different types of organisms. Some organisms are bisexual and some organisms are unisexual. So if the individual is bisexual, that individual will be called as hermaphrodite. But if the individual is unisexual, that can either be male or it can be female. Okay, so to determine whether the individual is male or female, we use different types of methods. And these are some of the methods: chromosomal method, genic balance theory, haplodiploid method, environmental method, etc. Or there are some other methods also. But as per NCERT, we will discuss only about haplodiploid method and chromosomal method. Okay, so let's talk about the chromosomal method. So before understanding chromosomal method, you should remember one thing, that sex chromosomes are of four types: W, X, Y, Z. W and X, these are the sex chromosomes which are responsible for femaleness, whereas Y and Z, they are responsible for maleness. When we write the alphabet in English, we write W, A, B, C, D like this, then W, X, Y, Z, right? And we know we give first priority to female, so they come first. So these are responsible for femaleness, and these two come later, and so obviously they will be male. Okay, so this is what you have to remember. Okay, now, so here autosomal set, if I write here AA, so you can see I have written AAA here. So A means autosomal set of chromosomes. So what do you understand by autosomal set of chromosomes? It means all the autosomal chromosomes. For example, in case of human being, chromosome 1, chromosome 2, chromosome 3 like this, 22 is present. Chromosome 1, 2, 3 like this, 22 is present. And in case of me, one X chromosome and Y chromosome is present. So I will say this is one autosomal set, this is another autosomal set. So with respect to human being, I will say from chromosome 1 to chromosome 22, I will call all of them as a or as capital A. Clear? Now, so XY method, ZW method, XO method or X0 method and Z0 method, these are the different chromosomal methods. So what do you understand by XY method? As per XY method, the individual may have AA plus XX chromosomal combination, or it may have AA plus XY chromosomal combination. That means 2 autosomal set and 2 X chromosomes, or 2 autosomal set and 1 X chromosome, Y chromosome will be there. Okay, so these are the chromosomal combinations. So if an individual has 2 autosomal set and 2 X chromosome, that individual will be female, because we know X chromosome is responsible for femaleness and here only X is present, so the individual will be female. Okay, so if this individual is female and it has XX chromosome, so the gamete will be of one type. Okay, so I can write here 2 autosomal set is there and 2 X chromosome are there. So when gamete is produced from this cell, one autosomal set will come and 1 X chromosome will come. So only one type of gamete will be produced. It will have 1 autosomal set and 1 X chromosome. That is what I have written over here, and this is represented as an haploid value. Okay, and if the individual is having 2 autosomal set and 1 X chromosome and another Y chromosome, then I will say that individual will be male and it will produce 2 types of gametes. One gamete will contain 1 autosomal set and 1 X chromosome, and another gamete will contain 1 autosomal set and another Y chromosome. Okay, so this is one gamete, this is another gamete, and both of them, they will have same chromosome number and they will be haploid in nature, NN. Okay, and because in this gamete X chromosome is present and in this gamete Y chromosome is present, that is why these gametes are of different types, and that is why I will say this male is heterogametic. Why? Because it is producing 2 different types of gametes. Okay, but the number of chromosomes in this male gamete and the number of chromosomes in this male gamete are same. Okay, that is why I will say, no doubt in XY method the male is heterogametic. The male is heterogametic, but the male gametes are having same number of chromosomes, and this XY method is applicable in organisms like Drosophila and human and Melandrium. Melandrium is a plant. And when I talk about this ZW method, this is just opposite to the XY method. Z is responsible for maleness. Okay, so if the individual is having 2 autosomal set and 2 Z chromosomes, okay, then the individual will be male and it will produce only one type of gamete, and that gamete will be having 1 autosomal set and 1 Z chromosome. Okay, and this will be haploid. Okay, and because this is producing one type of gamete, so this will be homogametic. But if the individual is having 2 autosomal set and Z chromosome, Y chromosome, that will be female. Okay, and it will produce 2 types of gametes. One will be having 1 autosomal set and Z chromosome, another will be having 1 autosomal set and W chromosome. Both gametes are having same number of chromosomes, both are haploid in nature. Because 2 different types of gametes are produced, this is heterogametic. So in case of ZW method, the female is heterogametic. ZW method, female is heterogametic, but in case of XY method, male is heterogametic. And this method is applicable in birds. And in both these cases, XY and ZW method, in heterogametic conditions, the number of chromosomes in heterogametic condition is same. Okay, now let's talk about XO method. Which method? XO method. So in XO method, one individual will be having 2 autosomal set plus XX chromosome, and another individual will be having 2 autosomal set plus XO, X0. That means only 1 sex chromosome will be present, second sex chromosome is absent. So in this case, this individual will be female and it will produce only one type of gamete, and that gamete will be having 1 autosomal set and 1 X chromosome, and that will be represented as haploid value. N. But if the individual is having two autosomal set and only one X chromosome, that will be male and that will be heterogametic. One gamete will contain one autosomal set and one X chromosome. Another gamete will contain only autosomal set but no sex chromosome. Okay, so this is haploid and this is N minus 1. N minus 1. So you can see here the number of chromosome is something and here the number of chromosome is something. It is having one more X chromosome, one chromosome extra, and this is having one chromosome less. So that is why this is male is heterogametic and the gametes are having different chromosome numbers. But in this case, you see, in case of heterogametic condition, in case of XY and ZW method, same chromosome number are present. Same chromosome number are present. Right? But in this case, the chromosome number is different. In this case, the chromosome number is different. Clear? And this method is applicable in grasshopper and Diascorea. Diascorea is also a plant which shows abnormal secondary growth. Now, what about Z0 method? If the individual is having two autosomal set and two Z chromosomes, that will be male, it will produce only one type of gamete. It will produce only one type of gamete, and the gamete will be having one autosomal set and another Z chromosome. Okay, and this is haploid. This is haploid. And if the individual is having two autosomal set and one Z chromosome and no other sex chromosome, then that individual will be female. It will produce two types of gametes. These are the two gametes. It is haploid and this is haploid minus 1, N minus 1. So here also the number of chromosomes in this heterogametic condition is different. That means in XO and X0 and Z0 method, in heterogametic condition chromosome number is different, but in XY and ZW method, in heterogametic condition the number of chromosome is same. Okay, and this Z0 method is applicable in butterfly. Now, let us talk about the other method of sex determination, that is haploid-diploid method. So in this case, the chromosome combination is not going to decide the sex of an individual. It is the ploidy level. If the individual is haploid, then it will be male. If it is diploid, it will be female. And this is applicable in honeybee. And in case of honeybee, 2n is 32. 2n is 32. So in case of honeybee, if the number of chromosomes is 16, that means if it is haploid, that will be male, and the male in case of honeybee is drone. And if it is having diploid chromosomal set, that means it is having 32 chromosomes, it will be female. And the examples of female honeybee are queen and worker bee. Okay, so this is mother, this is father. Okay, and they are crossed with each other. They are mated with each other. So when they are mated with each other, they will produce gametes. The mother will produce gamete. Okay, these are the gametes and these are basically eggs, female gametes. Okay, and because this mother is diploid, that is why it will undergo meiosis to produce gamete. And the process of formation of gamete is gametogenesis. Okay, so in case of mother, the gametogenesis is due to meiosis. It will produce female gametes. So these are the female gametes or eggs. Okay, but the father, being haploid in nature, it cannot undergo meiosis. It will undergo gametic mitosis and it will produce the gamete, and this is sperm. This sperm will fuse with the female gamete egg to produce the diploid cell, the zygote, and that zygote will develop into the daughter. Okay, so this will have 32 chromosomes. That means this will be 2n. Okay, so here what is happening? Fertilization is taking place. Fertilization is taking place. Fertilization is taking place. Clear? But this is also an egg, and if this egg does not undergo any fertilization, rather it undergoes multiple mitosis and further differentiation to produce new individual, that will produce the son. That means if the egg fertilizes with the sperm, it will produce the daughter. And if the egg does not undergo any fertilization but develops into new individual, that individual will be haploid, and that will be son. And that process of development of an individual without fertilization is called as parthenogenesis. Okay, now this son will produce gamete by mitosis and this daughter will produce gamete by meiosis. So these are the female gametes. This male gamete will fuse with the female gamete to produce the granddaughter, and this female gamete without fertilization will undergo parthenogenesis to produce the grandson. So from here you can clearly see, okay, let me write here what is happening. Fertilization is taking place. So male gamete has only one choice. It has to, the male gamete undergoes fertilization, the male gamete undergoes fertilization. But the female gamete has two options. It may, the female gamete may undergo fertilization, it may undergo fertilization, or it may, the female gamete may undergo parthenogenesis. Parthenogenesis. Clear? And one more thing. Can I say that the father has son? No, I cannot say. Why? Because this son is developed from this egg. And this egg comes from mother. Right? Because this son is derived from this egg. And this egg comes from the mother. And that is why this son has nothing to do with this father. This father does not contribute anything towards this son. That is why I will say father cannot have son. I repeat, this son is derived from this egg and this egg comes from the mother. That means this son has nothing to do with this father. That is why I have written father has no son. Father has no son. Clear? But the father has daughter. Why? Because this daughter is derived from the is obtained from the fertilization of female gamete and male gamete comes from the father. That is why I will say father has daughter. Mother has son as well as daughter. Clear? Now, this father has daughter and from this daughter grandson is produced. So I can say father has grandson. Father has grandson. So I will say father has daughter, grandson, but no son. Father has granddaughter also. Father has granddaughter also, but no grandson. But no son. Okay, that is what I have written. Father has daughter and grandson, but no son. Clear? Now, let me tell you something about mutation. But before discussing mutation, let me take some rest for 5 minutes and I will discuss about that. Now let's talk about mutation. Okay, but before discussing mutation, let me tell you one more thing, okay, which I have left here, that I should talk about some terms and polygenic inheritance. Okay, so sometimes we observe that one gene controls one character, and that phenomenon is called as monogenic inheritance. Mono means one. Here genic comes from gene. So one gene is controlling one character. So this is monogenic inheritance. And this monogenic inheritance is also called as qualitative inheritance. What do you call this one? Qualitative inheritance. Sometimes we observe that more than one gene control one character. And that is called as, more than one means many. For many I will write poly. So I will write this one as polygenic inheritance. And this polygenic inheritance is also called as quantitative inheritance. This is qualitative, this is quantitative. One gene, one character, monogenic. More than one gene, that means multiple genes, one character, polygenic inheritance. But some cases, one gene will control more than one characters. That means that will produce different types of phenotypes. And that is called as pleiotropism. What do you call that as? Pleiotropism. We will discuss polygenic inheritance and pleiotropism. Then we will talk about the mutation. Okay. Now, so let's talk about pleiotropism. This is a phenomenon in which one gene controls multiple phenotypes, more than one characters or more than one phenotypes. I can take examples like phenylketonuria. We will talk about this in detail later on. And in case of pea plant, a single gene affects or controls two different types of characters. One is seed shape, another is starch grain size. Because a given gene, one gene, is controlling in pea plant two characters. One character is this one, another character is this one. That is why there is an example of what? Pleiotropism. Okay, and that gene is B. It has two alleles, capital B and small a. So in case of pea plant, the genotype can be capital B, or capital B, small b, or small b, small b. In this case, capital B is responsible for round seed and small b is responsible for wrinkled seed. So here, because I have only capital B, so the seeds will be round. Here, because capital B is present, the seed will be round, and because here small b is present, so the seed shape will be wrinkled. Seed shape will be wrinkled. So I can say, with respect to seed shape, the capital B and small b, they show dominance. That means with respect to seed shape, the capital B is dominant over small b. Clear? But what about the starch grain size? Capital B is responsible for large starch grain and the small b is responsible for small starch grain. Okay, so here the starch grain will be large in size and here the starch grain will be small in size. And in this case, capital B, small b is present, and in this case the size is not large, it is intermediate size. That means with respect to starch grain size, capital B is not completely dominant over small b. That means I can say, with respect to starch grain size, capital B and small b, they show incomplete dominance. That means dominance is not a feature of capital B here. That means dominance is not a feature of the allele. Whether the allele will be dominant over another allele, it will depend on the character we take. The character we take. Okay, just a minute, the voice is cracking, I think, so let me change the battery. So clear? So what I'm trying to say here is that this, whether the capital will be dominant or not, it is depending upon the character we are considering. And that is why we say, the NCT says that dominance is not an autonomous feature of gene. Whether the allele will be dominant or not, that depends on the character we take. Okay, so that point you please note down. Dominance is not an autonomous feature of gene, rather it depends on the character we consider. For example, in this case, capital B is dominant over small b if the character I take is seed shape, and if I take starch grain size, in that case capital B is incompletely dominant over small b. So please write down that point. Dominance is not an autonomous feature of gene, rather it depends on the character we consider, the phenotype we consider. Okay, now what are the differences between monogenic inheritance and polygenic inheritance? In monogenic inheritance, one gene controls one character, whereas in polygenic inheritance, I don't know why the voice is having some problem, but I'm sorry for that. But in case of polygenic inheritance, more than one gene will control one character. Monogenic inheritance is called as qualitative inheritance. Polygenic is known as quantitative inheritance. In case of monogenic inheritance, the phenotype is normally not affected by environment, and if it is affected, it will be less affected. But in case of polygenic inheritance, the phenotype is normally affected by the environment. For example, let me explain you with the help of one example. If I take the flower color of pea plant that shows monogenic inheritance, okay, so the flower color the flower color of pea plant in Rajasthan will be identical to the flower color in Odisha or Andhra Pradesh. So the flower color is not dependent on the environment, it is completely dependent on the genotype. Okay, but if I take one individual and if I keep him in down south, the color will be slightly dark, and if I keep him in north like Rajasthan, or if I keep him in Kashmir like that, the color will be slightly white. So here the skin color character is controlled by gene as well as environment. Clear? That is why I have written here. And in this case, the variation is in kind. That means, and here the variation is in degree. What do you understand by this? So here, let me give you the same example again. When I say flower color is purple or white, that means the flower color is either completely purple or white. Okay, the purpleness of the flower in Rajasthan will be equal to the purpleness of the flower in the Madhya Pradesh. There will be no difference. There is difference in the kind. Okay, whether it is going to be white or purple. Okay, but if I take the skin color in quantitative inheritance, there is variation in degree. That means a person will be slightly dark, more dark, slightly white, more white, like this. So there is a degree in the skin color, whiteness or darkness. Okay, or I can take another example. If I take pea plant, the height of the pea plant is an example of monogenic inheritance. In that case, the height will be either dwarf, that means 1 meter, or 6 to 7 meter, or tall. No intermediate phenotype will be observed, 1 meter or 7 meter. But in case of quantitative inheritance like human height, individual with 1 feet is observed, 2 feet, 3 feet, 5 feet, 4 feet, 6 feet, 7 feet. So a number of phenotypes are observed. So there is a degree in the variation. There is a variation in the degree. How much tall, how much dwarf. But here either tall or dwarf. There is difference in the kind. Variation in the kind, either tall or dwarf. Here, how much tall, how much dwarf. It can be tall. I hope you are able to understand. And this shows discontinuous variation. Why? Because the plant is either 1 meter or 6 meter. But in this case, individual will be of this side, this side, this side, this side, this side, this side. It means you can see there is continuous variation, but there is no continuous variation. There is discontinuous variation. There is no intermediate value. There is no intermediate value here. A number of intermediate values are present. Okay. Now, in this case, in monogenic inheritance, normally intermediate phenotypes are not observed. But in this case, a number of intermediate phenotypes are observed. Number of intermediate phenotypes are produced. Okay, and in case of polygenic inheritance, the genes which are involved are called as polygenes. And in polygenes, the alleles can be dominant or recessive, and the dominant alleles of the polygenes are called as contributing alleles, and the recessive alleles are called as non-contributing alleles. The contributing alleles will contribute towards the phenotype, and all the contributing alleles of the different genes of polygenes, they will contribute equally. Okay. Now, if the number of polygenes is 2, then the F2 phenotypic ratio will be 14641. And if the number of polygenes is 3, the F2 phenotypic ratio will be 116, 16, 15, 20, 15, 6, 1. So what is the total number of F2 phenotype? I can use one formula here, and the formula is 2n plus 1, where n is number of polygenes, okay. So here, because I am having 2 polygenes, so I will get 2n plus 1, that means I will get 5 different types of phenotypes. You can see, 5 different types of phenotypes I will get, and here I will get 7 different types of phenotypes. How many? 7 different types of phenotypes. Clear? So let me ask you one question, that if the height of an individual with genotype capital A, capital A, capital B, capital B, capital D is 60 meter, and the height of individual plant with small a, small b, small b, small b, small d, small d is just a minute. Suppose this is 80 meters. How much? 80 meters. And suppose its height is 20 meters. So what will be the height of capital A, small b, small b, capital D, small d? What will be the height of this plant? Okay, this is very easy. Let me solve this for you. So you can see how many contributing alleles are there? 6 contributing alleles are there. Here no contributing allele. That means minimum height will be 20. Minimum height will be 20. And when 6 contributing alleles are present, how much is contributed extra? 60 is contributed extra. 80 minus 20 means 60 is contributed extra. And the contribution by A will be equals to B, and B contribution by A will be equals to D also. And how much it will be contributed? 60 upon 6. That means every contributing allele will contribute 10 meters. Everyone will contribute 10 meters. Okay. So here you can see what is the genotype given here? Capital A, small b, small b, capital D, small d. So how many contributing alleles are there? 1 and 2. So the minimum height will be 20 plus how many contributing alleles are there? 2 contributing alleles are there. So I will write 2 into 1 allele is contributing 10 meters. So it will be 20. So 10. So it will be 40 meters. It will be 40 meters. Clear? Now let us talk about the mutation. So what do you understand by mutation? Mutation is nothing but the sudden inheritable changes in the genetic material. So do you think that every mutation is inheritable? No. If the mutation takes place in the somatic cell, which is called a somatic mutation, that is not inheritable. That does not get transmitted from the parent to the progeny. But if the mutation takes place in the germ cell, which is germinal mutation, that is inheritable. But still we say that mutation is nothing but the sudden inheritable changes in the genetic material, that is DNA. Okay, so mutation in simple terms I can say change. The change is mutation. So in mutation, the change can take place in the chromosome structure or chromosome number or the base pair sequence in the DNA or nucleotide sequence in the DNA, nucleotide pair sequence in the DNA. So if the change in the during the mutation takes place in the chromosome structure, that mutation is called as chromosomal aberration. But if the change during mutation takes place in the chromosome number, that mutation is called as genomic mutation. And if the change during mutation takes place in the base pair sequence or nucleotide pair sequence of the DNA takes place, that is known as gene mutation. So where that change is taking place, depending on that, we classify mutation into 3 groups: chromosomal aberration, genomic mutation, and gene mutation. In gene mutation, either one or multiple base pairs are deleted, which is called as deletion, or substituted, which is called as substitution. Or added, which is known as addition. In case of chromosomal aberration, a segment of chromosome can be deleted, or can be duplicated, or can be inverted, or can be transferred from one chromosome to other, which is called as translocation. So these are the different examples of what types of chromosomal aberration. In case of genomic mutation, either the entire genome, that means all the chromosomes number, will change, which is known as euploidy, or less than the complete genome will change, that is known as aneuploidy. If the number of if all the chromosomes they show change in chromosome number, that is euploidy. If some chromosomes number change but not all chromosomes number change, that is known as aneuploidy. Euploidy is of two types. One is haploidy, another is polyploidy. Suppose there is an individual that is diploid, that means every chromosome is present twice, and if you lose one copy of every chromosome, that diploid individual will become haploid. And if you increase the chromosome number of every chromosome, that will become polyploid. And polyploid is again of two types, autopolyploid and allopolyploid. If the increase in the chromosome number is due to increase in the same genome, that is autopolyploid. If the increase in the ploidy level is due to the increase in the number of different types of genome, that is known as allopolyploid. For example, if I write capital A, okay, so this is diploid in nature. Here A is genome. So if AA becomes triple A or 4 times A or 5 times A, this is an example of autopolyploidy. If A is genome, and if I write AA, that means diploid, and if it becomes A, that means haploid, this is an example of haploidy. If one individual is capital A, another individual is capital B, that means here A, B are genomes, not genes, they are genomes. Okay, so from here I will get capital A, from here I will get capital B. So I'm getting two genomes, and these two genomes, they will combine with each other. Okay, so let me write this here. Okay, this is two genomes, not two genes. This is two genomes. From here I will get genome. From here I will get another genome. They will get fused with each other, so I will get two genomes here. And if they multiply, I will get capital A, capital B. So this was diploid. But this is tetraploid, and this tetraploid is due to increase in the copy number of genome of different genomes. Here genome A, genome B, two different genomes have come together. This is allopolyploidy. Clear? And what about aneuploidy? In case of aneuploidy, I can take some examples like monosomy. So in case of monosomy, every chromosome is present twice except one. Except one chromosome, every chromosome will be present twice, and one chromosome will present only once, and that is written as 2n-1. Okay, double monosomy is written as 2n-1 - 2. That means two chromosomes are present, are having one copy, whereas other chromosomes are having two copies each. Okay, nullisomy is written as 2n-2. Trisomy is written as 2n+1. Okay, double trisomy is written as 2n+1+1, and tetrasomy is written as 2n+2. Okay, to explain this, let me write here. This is very simple. Okay, let me take one example. Suppose one individual is having 6 different types of chromosomes. So these are different chromosomes. So every chromosome is present twice. And when every chromosome is present twice, I can write this condition as 2n. Okay, now if I add one chromosome here, chromosome 4. So every chromosome is present twice, but this chromosome is present thrice. How many chromosome I have added? One chromosome I have added. Which chromosome? Chromosome number 4. Because I have added one more chromosome, how many chromosome I have added? More? 1. That is why I will write 2n plus 1. Okay, here every chromosome is present twice. This is present twice, this is present twice, this one present twice, this one present twice, this one present twice, but this one is present thrice. So this is called as trisomy condition. Which condition? Trisomy condition. Clear? Now, what about monosomy? This is diploid condition. Every chromosome is present twice, but here I am removing one chromosome. So here every chromosome is present twice, but this chromosome is present only once. How many times? Only once. For 1 I will write mono. So this is an example of monosomy. So I have removed one chromosome. When I add, I will use plus sign. When I remove, I will use minus sign. So I will write minus 1. This is monosomy. Clear? What about nullisomy? What about nullisomy? So here I am removing 2 chromosomes, 2 copies of chromosomes. That means I have removed 2 copies of chromosome 4. So I can write here every chromosome is present twice, but chromosome 4 is present 0 times. And because this chromosome 4 is present 0 times, so I will write this one as nullisomy. How many chromosomes I have removed? 2 chromosomes, 2 copies of chromosome 4 I have removed. So I will write this 2n minus 2. So this is nullisomy. Clear? Now what about okay, so this is 2n. So what about tetrasomy? So let me add 2 copies of chromosome 2, or let me add 2 copies of chromosome 3. So before adding 2 copies of chromosome 3, this was 2n. So if I add 2 copies of chromosome 3, this chromosome 3 is present 4 times. So this will be called as tetrasomy. How many copies I have added? 2. So I will write this one as 2n plus 2. So this is tetrasomy. This is tetrasomy. Okay. What about double trisomy? So this is an example of what? Trisomy. So I will write this one as 2n plus 1. And if I add one more copy of chromosome 5, suppose. So this is also an example of what? Trisomy. So how many trisomy? Double trisomy. So for this one I will write 2n plus 1. For this one I will write plus 1. So this is double trisomy. Double trisomy. Okay. Now, if this is 2n, this is 2n. Now if I remove one copy of 3, so I will write this one as 2n minus 1. This is monosomy. Now if I remove one copy of chromosome number 6, so this is also, for this one I will write 2n minus 1. So this is monosomy, this is monosomy. So this is double monosomy. This is double monosomy. Clear? Suppose, okay, suppose I remove one copy of chromosome 3. So I will write this one as 2n minus 1. Clear? Now if I remove if I add one copy of chromosome 5, so I will write plus 1, so this is 2n minus 1 plus 2. Okay, so this is all about different types of what? Different types of aneuploidy. Different types of aneuploidy. And this aneuploidy is due to non-disjunction of chromosome. That means when the chromosomes fail to separate during meiosis, this happens. Okay, that is what I am going to write here. Just a minute. Aneuploidy is due to non-disjunction. Non-disjunction means non-separation of chromosomes. When the chromosomes fail to separate, aneuploidy takes place. Okay. Now, what about genetic disorder? The genetic disorders are classified broadly into two groups. One is chromosomal disorder, second is Mendelian disorder. Okay, so what is the difference then? If the genetic disorder is due to the gene mutation, if it is due to gene mutation, it is called as chromosomal disorder. Sorry everyone, very sorry, very sorry. Just a minute. If the disorder is due to gene mutation, that is called as Mendelian disorder. That is called as Mendelian disorder. And if the disorder, genetic disorder, is due to genomic mutation or chromosomal aberration, that is called as chromosomal disorder. Due to if the disorder is due to change in the chromosome number or change in the chromosome structure, that is genomic mutation or chromosomal aberration, that is called as chromosomal disorder. Examples I have written here: hemophilia, sickle cell anemia, color blindness, phenylketonuria, cystic fibrosis, thalassemia, myotonic dystrophy. These are the examples of gene mutation, Mendelian disorders. And Down syndrome, Turner syndrome, Klinefelter syndrome, Patau syndrome, Edwards syndrome. These are the examples of what? Chromosomal disorders. Clear? Now, let's talk about these disorders one by one. So when I say phenylketonuria, this is an autosomal disorder. Why? Because the gene which is responsible for this disorder is present in autosomal chromosome 12. That is why this is autosomal disorder. Okay, and in normal case, when the individual is having normal gene for encoding the phenylalanine hydroxylase, if the gene is normal or dominant, in that case, this enzyme will be produced, phenylalanine hydroxylase. This enzyme will be produced, and this enzyme converts phenylalanine amino acid into tyrosine. Okay, if the gene is in dominant form, normal form. Suppose that gene is A, so it will have two forms, capital A and small a. The gene for phenylalanine hydroxylase, suppose it is A, so it will have two forms, dominant and recessive forms. So if the genotype of an individual is capital A or capital A small a, in both of these cases this enzyme will be produced. And if this enzyme is produced, then this phenylalanine will be converted into tyrosine. But if the individual is small a, in that case this enzyme will not be produced. So no phenylalanine hydroxylase. Okay, no phenylalanine hydroxylase is produced. And if there is no phenylalanine hydroxylase, so this reaction will not take place. And if this reaction is not taking place, that means there will be accumulation of phenylalanine hydroxylase. That means in the body this will be accumulated, and later on what will happen, this accumulated phenylalanine will get converted into phenylpyruvic acid and other derivatives, and those chemicals will be accumulated in brain, and because of the accumulation of these compounds, there will be that individual will show mental retardation and reduction in hair and skin color pigmentation. Okay, so this is what happens in phenylketonuria. Clear? So you can see in phenylketonuria, I'm affecting only one gene, and when I affect one gene, it is affecting mental retardation, mental ability, and pigmentation. So one gene is affecting different types of characters or phenotypes. That is why this is an example of what? Example of pleiotropism. Clear? Now, what about color blindness? Color blindness is a recessive disorder, and the gene responsible for this disorder is present in X chromosome. That is why this is called as X-linked recessive disorder. And this is more common in males. About 8% of males. 8% of males they suffer from color blindness, and about 0.4% of females they show color blindness. That means in which the color blindness is more common, it is the males. Okay, now I have different types of genotypes. So if the individual is having XX, in that case the female will be normal. If the individual is XY, the male will be normal. And if the individual is XCY, male will be colorblind. If the individual is XCX, female will be colorblind. But if the female is XCX, it is normal but carrier. Normal but carrier. Clear? Now, what about hemophilia? Hemophilia is also an X-linked recessive disorder, and this is due to the defective blood clotting. That means the factors which are responsible for blood clotting, they are not produced, or they are less produced. They produce less. And this disorder was observed in the pedigree of Queen Victoria, and that is why this disorder is called as royal disease. And these are the different types of what? Genotypes. Okay, if the individual is written, if the female is XX, the female will be normal. If the female is XCX, XHXX, the female will be hemophilic, and this is very rare. Why this type of female is very rare? Hemophilic female is very rare because for an individual to be hemophilic, the father has to be hemophilic. If a female has to be hemophilic, her father has to be hemophilic, and a hemophilic person, a hemophilic man, cannot survive for such long till marriage. So this is why this is very rare. And if the genotype is XHX, the female is normal but carrier, and if the male is XHY, this is hemophilic male, and this will not survive for longer time period. If a small cut is given, it will die. Okay, now what about sickle cell anemia? Sorry, in case of sickle cell anemia, the structure of hemoglobin is affected. The quality of the hemoglobin is affected, and that is why we call sickle cell anemia as qualitative disorder. Whereas in case of thalassemia, the quantity of hemoglobin is affected, and that is quantitative inheritance. That is, sickle cell anemia is qualitative because the quality or the structure of hemoglobin is affected, whereas in case of thalassemia, the quantity of hemoglobin is affected. Okay, and the sickle cell anemia is autosomal recessive disorder. Why? Because in this case, the gene which is responsible for sickle cell anemia is present on autosomal chromosome. Autosomal chromosome. Clear? In sickle cell anemia, the hemoglobin is affected, and we know hemoglobin has two components, hem and globin, and the globin is composed of 2 alpha chains and 2 beta chains. And in case of sickle cell anemia, the gene responsible for producing this beta chain gets affected. It gets mutated. Okay, and in case of sickle, I will discuss this later on. Let me give some time. And this sickle cell anemia is due to point substitution mutation. What do you understand by point mutation? Any mutation in which there is a change in single base pair, whether that is deletion, addition, or substitution. If there is change in single base pair, that is point mutation. And in case of sickle cell anemia, the point mutation is due to substitution. So I will call this as substitution point mutation. Okay, this is responsible for sickle cell anemia, and this takes place in the gene responsible for beta chain, producing beta chain. Okay, and if I write the gene responsible for producing beta chain as capital gene as A and A, capital N and small a, so I can have 3 different types of genotypes, 1, 2, and 3. Capital A is responsible for producing normal hemoglobin, and this normal hemoglobin in case of adult is written as HbA. This A is adult hemoglobin, normal adult hemoglobin. Okay, and if the hemoglobin produced is sickle cell hemoglobin, that is written as HbS. And if the hemoglobin is produced is found in fetus, that is not written as HbF. We do not have to consider about this one. So when I write HbA, that means this is normal adult hemoglobin. This is sickle cell hemoglobin. And here here the A which I have written is the gene, the dominant allele, and small a is recessive allele. Dominant allele is present, so HBA will be produced. Dominant allele is present, so HBA will be produced. So this is normal male, normal female. Capital A is present, so normal hemoglobin will be produced. Small a is present, so sickle cell hemoglobin will be produced. Okay, why? Because see, this will produce normal beta chain, this will produce mutated beta chain, and if this normal beta chain combines with alpha chain, it will produce HBA, and if this mutated beta chain combines with mutated beta chain, it will produce HBS. Clear? And this individual will produce sickle cell hemoglobin. Okay, and this dies before maturity. Before maturity. Clear? Now, so this is one gene which is responsible for producing beta chain. So this is normal beta gene. Okay, so before here a number of base pair sequences are present. Here a number of base pair sequences are present. So suppose this is 5' end, this is 3' end, 5' end, 3' end, 5' end. Okay, so this is 3' end, 5' end, 3' end, 5' end, and this is 3' end. Okay, this is 5' end and this is 3' end. So this is normal beta chain which is going to produce the beta chain gene, which is going to produce the beta chain of the hemoglobin. So suppose I induce a mutation in this beta chain, and in this case this AT is substituted by TA. This AT is substituted by TA. So I'm doing the substitution mutation in one base pair. That is why this mutation is substitution mutation, point mutation. Okay, now this mutant beta gene, this is now mutant beta gene. This mutant beta gene will undergo transcription. It will undergo transcription and it will produce mRNA. Okay, and this picture, this is basically 6th codon. Which codon? 6th codon. And this GUG codon codes for valine. GUG codon codes for valine. Okay, so this is mutant beta polypeptide gene, and this will produce the sickle cell hemoglobin. Now this is normal beta chain gene, and if I allow this beta chain gene to undergo transcription, it will produce this mRNA, and this one is the 6th codon, and this 6th codon, if the 6th codon is GAG, it will code for glutamate. Okay, and this will produce the normal beta chain, and this normal beta chain, when it combines with the alpha chain, it will produce normal hemoglobin. So you can see this is valine is the first amino acid, histidine is the second amino acid, leucine, threonine, proline, valine, glutamate. Okay, here also the same sequence is present except 6th position. In this case 6th position valine, in this case 6th position glutamate. That means I can say in normal beta chain at 6th position glutamate or glutamic acid is present. In sickle cell hemoglobin beta chain valine is present. That means I can say in sickle cell hemoglobin valine substitutes the glutamate at 6th codon, at 6th position. Okay, now what about thalassemia? In case of thalassemia, the quality of the hemoglobin is not compromised, it is the quantity of hemoglobin is compromised. Okay, and that is why this is called as quantitative disorder. Sickle cell anemia is qualitative disorder. Okay, and this is of two types: alpha thalassemia, beta thalassemia. Delta is also present, but I am not going to discuss that. In case of alpha thalassemia, the synthesis of alpha chain is affected, whereas in case of beta thalassemia, synthesis of beta chain is affected. In case of alpha thalassemia, there is decrease in the synthesis of beta alpha chain, and when there is decrease in the synthesis of alpha chain or no synthesis of alpha chain, what happens? There will be increase in the beta chain synthesis. Only beta chain will be formed, no alpha chain is formed. And when only beta chain is formed, in normal hemoglobin, 2 alpha combines with 2 beta and they form the globin, and that globin combines with the heme to produce the normal hemoglobin, that is HbA. But in this case, no alpha is produced, and because there is no alpha, that is why all the beta, the 4 beta will combine with each other and that will form the beta 4. Okay, and when beta 4 is present, that globin when combines with heme, that will produce the HbH, and this HbH shows abnormal oxygen dissociation curve as compared to normal hemoglobin. Clear? Let me repeat here. In case of sickle cell anemia, the quality or the structure of the hemoglobin is affected. That is qualitative disorder. But in case of thalassemia, the quantity of the hemoglobin is affected. That is quantitative disorder. If the alpha chain synthesis is affected, alpha thalassemia. If the beta chain synthesis is affected, that is beta thalassemia. And in case of alpha thalassemia, there is decrease or no synthesis of alpha chain. In that case, what happens? There will be increase in the beta chain synthesis, and beta 4 will be formed. And when beta 4 is formed, that means the hemoglobin formed is HbH, and that HbH shows abnormal oxygen dissociation curve. And in case of beta thalassemia, there is either no synthesis of beta chain or less synthesis of beta chain. And whether there is no or less synthesis of beta chain, there will be increase in the alpha chain synthesis. That means there will be more synthesis in alpha chain. In beta alpha thalassemia, there is more synthesis of beta chain. In beta thalassemia, there is more synthesis of alpha chain. In this case, beta 4 is formed, but in this case, alpha 4 is not formed. So what this alpha chain is going to do? This alpha chain will go and bind to the plasma membrane of the RBC, and that will cause the rupture of the RBC plasma membrane. So ultimately, that will cause the damage of the RBC or death of the RBC. Okay. So this is thalassemia. Now, let's talk about Down syndrome. Down syndrome is basically an autosomal disorder. Okay, and this is due to chromosomal change in chromosomal number, that is genometric mutation, and this is due to trisomy of chromosome number 21. So because chromosome 21 is present twice, that is why the total number of chromosomes in that individual will be 47. Okay, every chromosome will be present twice, but the chromosome 21 will be present 3 times. And because chromosome 21 is present extra, that is why I have written plus 21. That means one more copy of chromosome 21 is present. And this was for the first time described by Langdon Down. That is why this syndrome is named as Down syndrome in the year 1866. And these are the different features of the person suffering from the Down syndrome. And I have told you just before some time that aneuploidy is due to non-disjunction of chromosome. That is why this Down syndrome is due to non-disjunction of chromosome number 21. This person will have short stature, that means it will have short height, small round head, furrowed tongue, partially open mouth, broad palm, many loops on the fingertips, big and wrinkled tongue, physical psychomotor and mental development is retarded. So these are the different features or characteristic features of the individual suffering from the Down syndrome. What about Turner syndrome? Turner syndrome is also due to non-disjunction of chromosome. That means this is also an example of aneuploidy. But in this case, the chromosome which undergoes aneuploidy or non-disjunction is chromosome X. So that is why I will say Turner syndrome is due to non-disjunction of chromosome X. Okay. So or chromosome X and Y, chromosome XX or chromosome X or Y, I can say. But that means I should say this is due to the not this Turner syndrome, even the Klinefelter syndrome, they are due to non-distinction of X and Y chromosome or X and X chromosome. That means due to non-distinction of sex chromosome, I should say. Turner syndrome is observed in females. In normal female, the total chromosome number is 46 and 2 X chromosomes are present in normal female. But the female suffering from Turner syndrome will have 45 chromosomes. 45 chromosomes. Just a minute. Okay, just a minute. Sorry, actually I had got a call. Okay, so Turner syndrome is found in females. In normal female, the total chromosome number is 46 and 2 X chromosomes are present. But the female suffering from Turner syndrome has 45 chromosomes, and in that case 1 X chromosome is absent. Only 1 X chromosome is present. In normal female, 2 X chromosomes are present. In Turner syndrome suffering female, only 1 X chromosome is present. So I write XO, or I can say 44 plus XO. 44 plus XO, or I can say 45 comma XO, X naught. And this individual female is formed due to fusion of normal egg with abnormal sperm or normal sperm with abnormal egg. Okay, so normal egg will have 22 autosome and 1 X chromosome, and abnormal sperm will have 22 autosomal chromosome and no sex chromosome. Okay, and normal sperm will have in this case normal sperm is having 22 autosome and X chromosome, and abnormal egg is having 22 autosome but no sex chromosome. So in both these cases, what is happening? 44 autosomes and 1 X chromosome but no second X chromosome. That is why that person will be female but no second X chromosome. That is why that will suffer from Turner syndrome. Okay, and that female will be sterile. Why? Because of rudimentary ovaries, and it will also lack other secondary sexual characters. Okay, now what about the Klinefelter syndrome? This is also an example of allosomal aneuploidy. Okay, Down syndrome is an example of autosomal aneuploidy, because in that case 21 chromosome shows non-distinction of chromosome. But in Klinefelter and Turner syndrome, the sex chromosomes, they show non-distinction. That is why both these are examples of allosomal aneuploidy. Okay, and in Turner syndrome, the individual which suffers is female. But in case of Klinefelter syndrome, it is the male which suffers from this syndrome. And in case of normal male, the total chromosome number is 46 and it will have 1 X and 1 Y. Right? Or I can say 44 autosomes plus X and Y. I can write like this, normal male. But in case of person suffering from Klinefelter syndrome, total chromosome will be 47. And 1 extra X chromosome is present. 1 extra X chromosome is present. Okay, so I can write XXY, or I can also write 44 autosome plus XXY. And this is formed due to fusion of normal sperm with abnormal egg, or fusion of normal egg with abnormal sperm. Okay, we know in case of what are the normal sperms? Normal sperm will have 22 autosome plus X or 22 autosome plus Y. These are the normal sperms. Okay, what about normal egg? 22 autosome plus X. This is normal egg. Normal egg. Okay, so this is normal sperm. Okay, and this is abnormal egg. This is normal egg, this is abnormal sperm. In both these cases, what will happen? I will get the 44 autosome plus XXY. Okay, that is what I am going to write here. So here I will get 44 autosome plus XXY. Here also I will get 44 autosome plus XXY. Okay, and such males are sterile, and these males will have some female features, like the voice, the female voice will be there, and the breast will develop, okay, which is known as gynecomastia. Now let us talk about the last part, that is pedigree analysis. From pedigree analysis the questions are not being asked, but please remember the few things. These are the different symbols, okay. If such symbols are given, that means that is indicating the male, and that male will be unaffected. And this is also male, but this male is affected or diseased. This is male unaffected, this is male affected or diseased. And this is female unaffected, this is female affected. Okay, this is individual with without having any specified sex. That means we do not know this sex of this individual, and this is unaffected. This is affected individuals with non-specified or unspecified sex. Okay, now this line indicates marriage, mating. Okay, these two lines indicate mating between relatives. Okay, and mating between relatives is known as consanguineous mating. Consanguineous mating. Okay, now these two are the diagrams. Which are given in the NCERT. Let me explain you this part. So when the diagram is given, you will be asked, this pedigree analysis shows which type of characters or disorders: dominant, autosomal recessive, X-linked dominant, X-linked recessive, like that. So for that I have some tips. One tip I will give you. Okay, so if the pedigree shows is of autosomal dominant, then you have to look at one feature, that in this case, if the parent is dominant but at least one individual is normal, that will be autosomal dominant. That means in this case, the key to identify whether the pedigree is autosomal dominant or not, here you should remember that the diseased or affected, you can say, diseased or affected parent may have normal child. Okay, so this is affected parent and this is normal child. Okay, so affected child is there, but normal child is also there. Okay, so I can say that is autosomal dominant disorder. Okay, so I can explain you here, no issues. Okay, so the affected individual can be homozygous or can be heterozygous. Okay, so because in this case the normal one is also present. Okay, because the autosomal dominant, so I will write here small a, and this will be capital A, small a. Why capital A, small a? Why not capital A, capital A? Because if the affected parent is autosomal homozygous, then all the progeny will be affected. Okay, and so how to can I write this is capital A or capital A, small a? To check this, to write this, I have to look at the progenies. Okay, if all the progenies are affected, it will be capital A. If some progenies are normal, that means it will be capital A, small a. Okay, so see, from here capital A will come, and from here small a will come. From here small a is coming, from here small a is coming. Okay, from here and here small a are coming, and this is small a, and from here capital A is coming, and from here small a is coming, and this is small a. Okay, now because both of them are having small a, so all of them will be small a. Okay, so this will be normal, and from here small a, so this will be small a, and here also small a, and from here capital A will come, and from here small a will come, so this will be diseased. So the key point here is, if the parent is diseased but normal child is also present, that will be autosomal dominant. And how to find out the genotype of affected individual? Okay, So the genotype of affected individual, in this case, affected individual genotype can be, it can be capital A, or it can be capital A, small a. Okay, when capital A is taken, if all its children are affected, if all its children are affected, then I will take capital A in case of autosomal dominant. And if some children or child are normal, then I will take capital A, small a. See, in this case, this is affected and some children are normal. Affected, some children are normal. That is why I have taken capital A, small a. Okay. Now, what about autosomal recessive? Here the key is if both parents are normal and they have diseased children. Okay, they have diseased children, along with they can have normal children also, no problem. But if both parents are normal and if they have diseased children, then that will be autosomal recessive. You can see normal but diseased, normal but diseased, so autosomal recessive. Okay, so in this case I can write here, this is capital A, small a, this is also capital A, small a, both are normal. This will be capital A. This can be capital A or capital A, small a. This will be small a, from here, and from here small a will come. Okay, this will be capital A or capital A, small a. Okay, and can I take capital A only? No, I cannot take, because here the diseased individual is present. So I will take capital A, small a only. Capital A, small a I will take, and this one will also be capital A, small a, and this one can be capital A or capital A, small a. This one can be capital A or capital A, small a. This one will be capital A or capital A, small a. This one is small a, like this. So in this case, let me write here one point. In this case, capital A, in this case I will write capital A is diseased, capital A, small a is diseased, small a is normal. This is normal individual. Okay, diseased individual. This is diseased individual. Okay, in this case I will write capital A, small a, small a. This is diseased. This is normal. This is normal and carrier. So what is the genotype of normal individual in case of autosomal recessive disorder? It can be capital A or it can be capital A, small a. So to find out whether the genotype will be capital A or capital A, small a, I have to look at its children or progeny. Okay, so if all of its progeny are normal, then I will take the capital A. Okay, so I will take this one. If it's all progeny, or I can write children, are normal. then I should say I should take capital A small a. And if some diseased children are present, then I should say I should take capital A small a. See, normal and normal, but it has diseased child. That is why I have taken capital A small a. Okay. So these are some of the few points. So hope and let me write last point. Let me explain you last point. Mendel experimented on pea plant and he took 7 characters. He took 14 varieties. Okay, and he performed this experiment for 7 years from 1856 to 1863, and his work was rediscovered in 1900 by 3 scientists individually, okay, separate independently, and the scientists who rediscovered Mendel's work were Hugo de Vries, Carl Correns, and Schermark. Okay, so this is another point. Okay, and let me write one more point here. Okay, 1 or 2 points here. So let me write some formula to find out the types of gametes. Okay, let me write here. N is for number of heterozygous condition. N is for number of heterozygous condition. Okay, so if I say types of gametes produced by an individual, how many different types of gametes will be produced by an individual? So for that you use a formula. And the formula is 2 to the power n. Okay, so if I write capital T, it will produce only one type of gamete. Why? Because heterozygous condition is 0, so I will write 2 to the power 0, so it will be 1. If I write capital D small t, it will produce 2 type of gamete because heterozygous condition is 1, so it will be 1. If I write capital R small r, capital Y small y, so it will one heterozygous, second heterozygous, so it will produce 4 different types of gametes. 4 different types of gametes. Okay, now what about the F2 genotypes? Types of F2 genotypes. For this, I use the formula p to the power n. Okay, so if I ask you individual with capital small a, small b, capital C, small d, how many F2 generations will be produced? How many F2 generations will be produced? So in this case, how many genotypes will be produced? How many genotypes will be produced? Okay, so in this case, here I will not take F2 generation. I will say if this individual is self, then how many genotypes will be produced? For this, I will use the formula 3 to the power n. Okay, so this is one heterozygous condition. This is another heterozygous condition. So it will produce how many genotypes? It will produce 3 to the power 2. So it will produce 9 genotypes. How many? 9 genotypes. Okay, so what about the phenotypes? What about the phenotypes? For phenotypes, I use the formula 2 to the power n. Okay, so here the phenotypes will be, this is heterozygous, so it will produce 4 different types of phenotypes. Okay, so these are some of the formula you have to remember. So I hope this was productive. Okay, and take care. Thank you.